Pak Arman, Prof. Asef, or other committee is Professor Aminul is already joined with us?
Yep.
Okay. I think he sent a chat room.
We have to see. I don't
Murshid Muhammad Mursid Hassan. Okay. Not yet, yeah? Please remind me if Prof. Aminul's already joined with our chat room, yeah? Okay. Thank you, Pak Arman, Pak Asep. Pak Arman, Pak Asep, Prof. Asep is already joined for Prof. Amino. Any information about our first lecturer?
Hello, Prof.
Yes,
Aminu
Prof.
already joined with us.
Okay, let's see.
Halo, Prof. Aminu.
Hello, Prof. Aminu. You already joined with us, and I think all the participants already joined, yeah, with the room, with a very heavy face, with empty stomach also after take lunch. Okay, yeah. Prof. Aminu. Hello, Prof. Aminu. Already join with us. Hello. I think Prof. Aminu is not join with us yet, yeah, Prof. Asep, Pak Arman. Okay, yeah. While waiting for the honorable speakers, I just only want to remind to all dear participants, as you can see on the slide, the rules of our during the lecture, yeah. You have to use appropriate outfit and also behave during the program, and you also have to put your complete name, yeah, with the format that already seen on the chat room. And also you have to turn or mute your audio, but please turn on your video, yeah. Hello, Prof. Amin. Prof. Aminu.
Yes, sorry, I was getting a trouble to join.
Okay, thanks God. Alhamdulillah, you already joined. Already joined with with us, yeah, Prof. Aminu. Thank you very much. It's a great honor for us that already joined, because all the participant is really eager in this room, waiting for your lecture actually, yeah. And here we already have Prof. Asep Gunawan as the committee. Prof. Asep, would you mind to say something to Prof. Aminu before we
Hello,
start?
Prof. Aminu. Thanks a lot. Thanks a lot. Yeah,
Thank you, Professor.
for giving us a chair today.
Okay,
I already got a bit late, 10 minutes probably. So, in fact, I'm not fully, I mean, a little bit sick actually, but I was committed. That's why I'm trying to communicate with you all. Let's see.
Yeah, thanks a lot. Yeah, hopefully you getting better soon.
Yes, I do hope while discussion with the participants, you have still a lot of energy, yeah, Prof. Amin.
Yeah, sure.
Okay. So we start, yeah. Okay, dear participant, thank you very much. Please welcome to our honorable speakers, Prof. Aminul Islam. Thank you for joining with us. This is a second day of our program, our summer school program. And at first, I would like to apologize if mine is not quite flat, yeah. We speak in easy English. So I do hope you can still capture the information that I say during this program. Yeah. Today, similar with previous day, we have a two honorable speakers with a different session, of course. And then the first one will be done by Professor Aminul Islam. Again, thank you very much, Prof. Join with us, sharing the knowledge, sharing the with us, even though we are in a virtual, yeah, in a virtual situation. And I do hope you are in a very well condition, even though that you said that you are not so well today. But I hope during this lectures, you will be uh, very well, yeah, in good condition. And also for the participant, I think we can see a lot of happy face here, and I do hope not to your stomach not to full, so still a lot of energy, yeah, listening to the lecture. Okay, the rules of the summer school programs is already presented by the team, yeah. That you have to turn on the video during lecture, but you have to mute the audio, yeah. And we'll have you can turn on the video or the audio during the discussion, or if the committee asking you to turn on the audio. Before we start, let me uh, introduce Prof. Aminu. I will share screen, yeah. Permission for to share the screen. So this is the profile of uh, our honorable speaker this afternoon, yeah. The complete name is Professor Dr. Aminu Islam. Already full professor, yeah, Prof. Aminu, since January 2021. Work at Department of Medicine, Faculty of Veterinary Science, Bangladesh Agriculture University, yeah. And this is the educational background of the Doctor of Veterinary Medicine in Bangladesh Agriculture University, and then have a double, yeah, in a master program, yeah, Prof. Aminu. First in Master of Science in Medicine in Bangladesh, and the second one is in Tropical Animal Health in Belgium, in Antwerp, Belgium, and then hold a PhD from University of Bonn. I think it's similar to Prof. Asep, yeah? Professor Aminullah from Bonn University. Okay, and this is the research interest of our honorable speaker, Prof. Aminu. So if one of the participants are interested in this field of research, so perhaps after this program, Prof. Aminu will give a time to communicate or something, yeah? Perhaps some of the participants would like to continue their master or PhD study. So perhaps Prof. Aminu will give a chance to all participants to discuss afterwards. And this is, we can see that how productive Professor Aminu for the publication. Let's see, and then this is many publication has been published by Prof. Aminu, yeah, regarding his field of expertise. And also this is contribution, not just only published, but also become editorial or reviewers in many scientific journals. Okay, that's all the short CV. I think your CV is very long, Prof. Amin. So I do apologize. The committee make it a little bit short for the for this. So you will have 60 minutes and hours for presentation, and then will be followed by 30 minutes of discussion. So is it do you prefer if we committed to help you during the presentation, or you will be done by yourself for the presentation, Prof. Amin?
It's okay. I can do it. I can share my screen. Then
Okay. Pak Arman, Do you already Prof Amin become a co-host or something? The co-host?
It's okay. You can
Okay.
better make me co-host.
Okay.
That's
Good.
fine.
Okay. I do hope dear all participants are enjoy and then with these lectures, yeah, from Professor Aminu, and I do hope a lot of new knowledge, insight about this topic. So, Prof. Aminu, an hour from now, times is yours. Please, thank you.
Thank you very much, Dr. Aditya. Let me just a second. I'm starting to share my screen. Uh, can you see my screen?
Clearly, Prof.
Okay, I'm here. So, first, I would like to thank the organizer, particularly my friend Professor Asif, to invite me to communicate with you all. And it's really a great honor to me to share some academic point view with you all. And yeah, it's a very terrific time, very pathetic and very I would say very tough time. All the world is passing nowadays due to this ugly COVID-19 pandemic. All the plans and all the things are changed in such a way that nobody can control it. So thanks Almighty God Allah that we could meet and we could survive. We survived and we could continue the academic and research in our site. So I would like to welcome you all the participants to my session here. So what Dr. Aditya spoke about me, I think it's he speak a little bit more. I'm not that much anyway. Thank you. Uh, let's start a bit about the topics today. I will discuss something about actually the epidemiological background of COVID-19 pandemic. In fact, now it's almost 1.5 years we are facing this pandemic situation, and there are 2 or 3 sometimes the COVID waves comes and going on, and a huge bunch of informations are available, particularly due to advancement of the internet facilities. Uh, mm, the informations about COVID-19 is a huge informations. So it's very tough, um, particularly sometimes to uh, um, pick the right informations for the academic purpose. This is huge bunch of information. So let's try to share some uh, academic informations particularly, and I guess many of you are somehow came across the informations what I will share today about COVID-19. But anyway, that some of you might be interested in, or let's just share. So in my talk, I will first talk about the pandemic history, and then I'll go for the a little bit biological or taxonomical informations of coronaviruses. Then a little bit about descriptive epidemiology. I mean, distribution, outbreak distributions, and epidemiological curve. Then I'll talk something about the epidemiological triad, that interactions of host, virus, and environment, and the preventive approaches, what we used to do for epidemiological perspective, then risk factors and impact of COVID-19, and so on. a little bit advice and a little bit lifestyle changes to cope up with this pandemic situations. So, in the history, uh, it's about 100 years back. There was a severe respiratory viral pandemic in 1980. It was due to avian influenza viruses, and it outbreaks start from the Spain. That's why it was termed as Spanish flu, and a huge mortality was recorded. It was an history of the world, and people never before experienced this sort of high mortality. And the pandemic word, in epidemiological perspective, we use the word pandemic when a disease occurs more than one country, the epidemic manner. That means more than normal scenario. In epidemiological context, we do classify the disease occurrence in four. That is, one is regularly occurrence, that we call endemic disease, and uh, if the disease occurs very rarely, we call sporadic. But if the disease happens so frequently, then we call this epidemic. And this so frequently happening, it comes over more than one country, then we call it pandemic. So the pandemic comes so frequent occurrence of a particular disease in more than one country, and the Spanish flu was spread all over the world, and then so many pandemics happening and so many human death recorded. Then comes this 2019, 2 years back, end of the 2019, December 2019, um, very first time at Wuhan province of China, people affected with pneumonia of unknown causes. Then end of the December 31st, the unknown cause, the death of this respiratory illness and respiratory illness follows by death. It was reported to WHO, World Health Organizations. Then January 7, 2020, this causal viruses of this unknown respiratory problem was identified as a coronavirus. As this coronavirus was a little bit different, not like any previously determined or identified coronavirus. That's why it's called novel coronaviruses. Then January 13, people affected with this novel coronavirus outside the China. That means it's transmit from China to other countries, started transmission. That is the nature of the pandemic. What we call what I just speak about, that so frequent occurrence and more than one country. Then March 11, 2020, WHO announced this respiratory illness and death due to novel coronavirus infection as a pandemic diseases, and they named the disease as COVID coronavirus disease, and the year was starting 19. That's why COVID-19. That is coronavirus disease 2019. That's why the disease named as coronavirus disease 19 or COVID-19. And this coronavirus, biologically, it belongs to the Nodoviridalis. And Nodoviridalis has three families: Coronaviridae, Roniviridae, and Artariviridae. Artariviridae and Nodoviridae are a little bit different, but the Coronaviridae again has two subfamily. That is Coronavirini and Torovirini. Torovirini also a little bit different biology. actually is causing this pandemic COVID-19 and also some other previous pandemic. Uh, the novel coronavirus that is detected in 2019 from Wuhan province, it was the causal agent of COVID-19 actually. But there are 7 other coronavirus strains that affect human being. And let me tell about, do you know, maybe coronavirus is a member of RNA virus family that I talked the last slide. The characteristics of coronavirus, this physical evidence in uh, electron microscope, it looks like a crown. Due to this crown shape, lipoproteins is called corona. That's why the name of the virus come this crown shape appearances of this virus particle under electron microscope. That's why it termed as coronaviruses. So this coronavirus is not new. Coronavirus has appearing from long before, but this one, what we are facing now, is a novel one. The previous one, some other strains happens before, and it also causes some other pandemics. So the now the present one, the coronavirus, and that is associated with respiratory syndrome, and actually acute respiratory syndrome, and the coronavirus type 2. That's why severe acute respiratory syndrome coronavirus 2, that is detected on the Wuhan infection 2019. That's why it's acronymed as SARS-CoV-2, SARS-CoV-2. The SARS-CoV-2 is the causal agent of COVID-19. What we told before is belongs to the beta genus of this coronavirus. And 7 coronavirus in total affecting the human being, but some other coronaviruses are affecting animal also. Even cattle, for example, beta alpha, beta 1, and we are facing the coronavirus problems in also poultry. So the major concern now is SARS-CoV-2 that is happening the present pandemic. But before, there was another coronavirus strain that is called MERS-CoV that also happened as a Middle East respiratory syndrome. Not like so severe as COVID-19, but it was also reported as a pandemic. Then another coronavirus pandemic happened before, that is also called it was 2003, the first coronavirus pandemic, SARS-CoV. That's why the present one is termed as SARS-CoV-2. The already has a previous one. So at least 3 SARS pandemic we are experienced with. We talked about the coronavirus strain. Actually, it looks like a crown. That's why it termed as coronavirus. But the important thing under light microscope is the spike proteins of this virus envelope. It's a protein structure, and it looks like a spike. That's why it named as spike protein. And this is the um, this is the structure actually what is responsible for the infections and entering to its particular host. And we'll come later how coronavirus get into the human body cell and how it create the severe pathogenesis. And you maybe know about the coronavirus genomic structure. So it's a genomic background, what we call spike proteins at the outer side. Just what we saw the last slide. then N protein, M protein, HE, that is hemagglutinating protein. These are all typical viral structure under light microscope. What makes it different is a spike protein, that is particularly the site that is helps to attach and get into the human host cell. Um, Coronavirus genome. Let's see this slide. Actually, this coronavirus, there was a myth about the coronavirus pandemic when it comes middle of the last year or autumn of the last year, that maybe the coming winter, that is December 2020 and January, February 2021. I mean, uh, the winter season might have severe or severe pandemic, or the winter season has strong predispositions of respiratory infections of these novel coronaviruses. But it has a controversy and a lot of debate over there. I'm not going on that. But what I'm talking now is uh, because of RNA virus structure. It's an RNA virus. That's why almost all RNA structure is more fragile than any DNA structure. Even it's a host RNA or DNA. Particularly, it's a viral RNA, and due to RNA structure, as RNA structure is more fragile and very vulnerable, that's why it changes its genetic material so rapidly that we called mutation. So changing the genetic materials in response to environment or other stimuli, it's the mutation. So mutation happens so frequently in all RNA virus, including these novel coronaviruses. That makes the control difficult. Genetic diversity of coronaviruses actually, when any sorts of pandemic happens worldwide, scientists are trying to get the genomic structures of these causal viruses affecting in different corners of the world. From the patients of different country or region, they isolate the RNA and sequences the whole RNA viral genome and try to summarize whether the particular source of RNA is similar or dissimilar with the RNA comes from another country or regions. And this is the dendrogram. What who we have seen now is the red one is human SARS coronavirus 1, though it's not 1, as because it's the first appearance of the pandemic. That's why it's named as human SARS-CoV. But human 2019 nCoV, that is the novel coronavirus, and later it termed as SARS-CoV-2, not directly related, but if we go back a little bit back, then there is a little similarities from the same dendrogram. And there is another myth whether this novel coronavirus come from a natural animal host like bat or others, through a secondary intermediate host, then comes to human being. Still, these things is not scientifically proved, but there are some evidence there might be a transmission route from bat to a pangolin than human being. So the respiratory, this coronaviruses particularly cause the respiratory infection. the normal human coronaviruses, that is not like this novel strain. Usually, what they cause the common cold and upper respiratory tract infection, sneezing, nasal secretions, and a little bit like a common cold, influenza, something like. Um, and most of the coronavirus strains are affected with the upper respiratory tract. And upper respiratory tract, when we comes or to face across any upper respiratory tract infections, our body immunity, particularly the innate immunity, has spelled out this respiratory infection. Usually we sneeze, and due to this sneezing or nasal secretions, the viral particle comes out, and it doesn't make so severe pneumonia or lower respiratory tract infections. I mean, the viral particles spelled out, even they get entered into the particular, the major respiratory organ, the lung. But in differently, the novel coronavirus, that is SARS-CoV-2, also other coronavirus that makes two pandemic, SARS and MERS. And this is not like other human coronaviruses, as they affect the upper respiratory tract. This novel one comes down, and from nostril they got oronasal infections and comes down, particularly infect the lung and lung alveolar cell. That is the danger and main cause of concern, that it affect the lung and cause severe pneumonia and subsequently severe pathogenesis, even death. This novel coronaviruses, how it comes to the alveolar cells, and there is there is a non-receptor, what you call SE2 receptor. It found in the lung alveolar cells, and when this virus comes inside, the spike protein of the coronavirus surface, it attaches to this ACE2 receptors of this alveolar cells, and it fused and get into the alveolar cells, and subsequently it causes the pathogenesis and severe diseases, particularly due to alveolar macrophage and alveolar cells and alveolar macrophage got infected, and number of alveolar cells and alveolar macrophage goes down, and because of these cells has a major contributions on immune system, and because of low number of alveolar macrophage, body immunity goes down, and it subsequently cause severe disease, even death. What we call the SARS coronavirus to the virus infections. Actually, the last slide, what we have seen, this is a little bit more elaborative one. What are the fate of these host virus interactions, particularly for these novel coronaviruses? This is lung alveolar space. This is upper respiratory tract, goes down the virus, and the virus interact with the alveolar cells through the spike protein and ACE2 receptor. And after binding these receptors and viral protein, the virus get inside. If the and after getting inside the virus particle, it start the replications and increase the number, and the virus life cycle, actually after getting into the host cell, it goes under rapid replication to increase the number, and after getting huge number, it causes the cell death, and after rupturing the cell wall, the viral particle, particularly the viral RNA, comes out and infect the another neighboring cells. But if the host immune systems are strong enough, then after getting this viral particle inside, and there is one host immune response is happening. We call this interferon response or interferon phenomena. If the alveolar cells initially response get infected with one or low doses of coronaviruses, and it get a little time, and this time within this short times the host, particularly the alveolar macrophage and alveolar epithelial cells, signals signals the interferon response, and this interferon comes out from the infected cells and it give a signal to neighboring healthy cell, and the healthy cell get immune to protect the subsequent infections with other coronavirus that is coming to infect that one. That's why if the immune systems of the particular host is strong enough, then it produces this interferon phenomena. And if the virus infections is a little bit lower dose, then body gets some time to create these interferon responses, and subsequently the next infection prevented and released from the cells, dead cells, and the host get recovered from the infection. On the other hand, if the host, particularly the human, if the host immune system is not strong enough, or the particular Roman is previously infected with other diseases, or even any non-infectious diseases he is suffering from, that is particularly affect the lung, or even the cardiac infections he has, kidney problem he has, that also affect the respiratory function. Um, Particularly if the host has low respiratory function, then if he got a higher doses of coronavirus infections and he didn't get enough time to produce this interferon phenomena, within the lung alveolar macrophage of his system, then his immune systems cannot fight against these viral infections, and ultimately he the whole lung is infected, and the alveolar cell is damaged, particularly in pulmonary alveolar damages. We know that pulmonary alveolar cells are the major roles is oxygenations, exchange of air, exchange of oxygens through uh, respiratory systems. And once this oxygen exchange systems is damaged or hampered, then the patient suffering from anoxia. The lung cells didn't get the oxygen from the blood, uh, and continue his breathing. That's why, due to anoxic shock, the patient goes to death. This is the bad or worst happening of coronavirus infection. This slide actually we talked about a little bit before, that there was a myth whether this SARS-CoV-2 originated from any sorts of wild animals, that is and wild mammals called pangolin, and through these pangolins it comes to human being. Some sort of evidence, research articles comes that pangolin might be a source, the origin of this novel coronaviruses. And how this novel coronavirus to transmit from infected animal to infected human, infected patient to other susceptible patient. This is big questions and million dollar questions we say, and this is particularly the major concern, the transmission route for any sorts of infectious disease. If the transmission cycle is complex and transmission happens is so fast, then it is then the virus or then the disease is more dangerous. What happens in this SARS-CoV virus infection transmission or spreading? Because this is one thing is novel virus. In fact, lower respiratory tract, this is a risk, and the disease causal agent, the virus is highly transmissible, and transmission occurs multiple routes, particularly in the respiratory infection. All sorts of respiratory infections are got the aerosol transmission, direct contact, droplet transmission, and so other transmissions are happening that got the primary infections through inhalation or the oronasal route. Either the infection get into through nasal or inhalations, or the little bit on mouth or other eye opening. All these goes, and there is a common anatomical structures is pharynx, then larynx, and goes down the respiratory systems. That's why if the all sorts of respiratory infection, the major route of infection is nostril or nose, nasal cavity, and a little bit on eye and the mouth, because there is a common passage of eye, nose, ear, and even the mouth cavity goes down. There is a common opening. Uh, this there are a lot of informations about how novel coronavirus is transmitted from infected patient to susceptible patient. One thing is respiratory droplet. If the patient got infected and his lung cells got infected, and when he exhale, then the infected virus particle comes out through his nostrils, and it makes a small droplets of nasal secretion, and this droplet, due to gravitational force, it comes down to any sorts of surface. And if by nature or by habit we used to touch our nostrils during sneezing or coughing, or any cleaning of any sorts of respiratory nasal secretions, then the droplet comes to the surface or our hands, and by nature or by mistake or by habit, if we touch this hand or unhygienic hand to nose, again, or eyes or mouth cavity, that is the vulnerable situations, and that is the actually the way how we get infected with this novel coronaviruses. That's why it is suggested that the respiratory droplet, when comes out from an infected patient, it um, usually comes down to the surface, neighboring surface, but it takes a little bit time, and it spread a little bit. It spread a little bit its surroundings, and how long it goes or how far it goes. That is a question. the expelled nasal secretions containing the infected viruses, how far it can go. It usually there are a lot of experiment over there, and scientists say that's 6 feet or minimum 3 feet, 3 to 6 feet distance. If if the two persons maintain, then the infected persons, even though he expel the coronaviruses through his nasal secretions, it may not come in contact to the neighboring susceptible person. Here is a picture you can see that's if the persons, infected persons, uh, the virus particle comes out from his nasal secretions uh, in the air, or it comes down through the uh, to the surface. And if a healthy susceptible person come closer to him and slowly closer and closer, even within this period, within this area, if he got exposed, then there is a chance to get infected from the infected patient. And another way, what we call fomite transmission, infected patient expel the viruses and through his hands or other um, clothing or whatsoever from the infected persons. If he touches any surface, and from the surface another healthy person is got infected, this is called fomite transmission. And another way is called airborne transmission. If the persons exhale the virus particle and it floated over the air and a little bit more distance, and particularly the compact room, the virus particles uh, remain in the air a little bit longer times, and the susceptible patients come in exposure, then there is a chance to get infected. This is the way. This is called airborne transmission. This is more dangerous, and information about airborne transmissions of COVID-19 has a recent idea and recent findings of the science. And from the very beginning, from the early of this pandemic, it was a question, or it was not clear whether um, this novel coronavirus is cause airborne transmission or not. But it is proven there. The latest Lancet paper explained that there is a chance of airborne transmissions of this SARS-CoV-2. That means a compact AC or closed door room has a higher risk of virus transmission than any open space or outside of the room. So a little bit outbreak investigation, epidemiological concept we say, particularly the principles of epidemiology. We studied the incidence, distributions, and determinants of any sorts of diseases in order to design a possible intervention strategy or control strategy. Therefore, understanding the outbreaks and mode of transmission and how it's distributed over the population is a key factor to make or to design a particular prevention or control strategy. What I said before, that a lot of information about COVID outbreaks are available online, but it's important to pick the real information or the correct information or authentic information. We considered the WHO website for the authentic information about this pandemic. And from the WHO website, there is a particularly add-on and dashboard of always updating the coronavirus information. Until now, it's 186 million people are infected with this coronavirus. This is actually the official confirmed case. The real scenario might far bigger number. What do you think? And the official number of death cases is even more than 4 million. So it is the highest number of infected cases and number of deaths in the history of pandemic. And the global scenario, what at a glance from this figure, from this EPICAR, we can see is the distribution pattern or the occurrence of this disease in over the time. From the very beginning, it was low number is very few, and the number goes up a little bit, fluctuating curve we have seen. Then in the December last year, November, December until January, the curve was peak. This is the global scenario. And in February, the curve goes a little bit down. There was a hope that this pandemic may be going over. But unfortunately, it happens, it comes up again, and particularly the March, April, and January, March, April, May. March, April, May, it was again have a second peak, we called second wave of this infection. And then end of the May and June, it goes a little bit down. But particularly in Bangladesh, my country is now we are facing, we call maybe it's third wave, and right now it's very bad scenario. Even yesterday we got the highest number of deaths in our country this pandemic. So we are facing the peak of this curve. Though the global scenario is improving nowadays, the same curve, most of the cases of death follow the number of cases, confirmed cases. But you can see that the death cases or death curve is a little bit flattened than the confirmed case curve, and the death curve had not that much clear peak, but 1 second clear peak is there. But the early months of the last year, it was a little bit flattened. Then December, November, December, January, again March, April, May, we got a little bit high. Here is a little bit clustering of the previous graph. This is clustered on the regional basis. The yellow one is America, then Europe, and the Southeast Asia is the purple one. If you see the purple curve, this is actually number of confirmed cases, and overall Asia Pacific situation goes very worse this year. Even from the last year. Last year it was not that much worse, but this year it worse than the previous year. This is also same for the number of deaths. Particularly the Asia Pacific region had little bit smaller experiences, smaller number of deaths, fewer number of deaths last year. But this year, particularly last few months, last 3 months, number of deaths in particularly the Asia Pacific region was highest. if you go back a particular country-wise curve, USA has a single peak and goes down. India has recently got the peak and now it comes down. Brazil, it's a zigzag curve and always goes up, goes down, and still the curve in the same manner. But Russia, it goes one peak, the second peak goes higher, then comes down and again up. France also has two peak. Turkey has a small and second peak. United Kingdom has only a single peak, but recently there is a little higher trend of this curve. Argentina also get bad experience in recent months. Colombia also bad experience in the recent months. But in Germany, if you say, if you look at the Germany, they experienced the two curves, but now the curve is goes down and continue. So this is the scenario we have seen how the outbreak is have been proceeding with, but how we can control the infection or the disease, particularly from the epidemiological point of view. Uh, there are several theorem of or causations of infectious disease and the preventive model for infectious disease. Among them, epidemiologic triad, a model that composed three components: agent, host, and environment. Over time, has been has been considered as the strongest and strongest model for designing the prevention and control strategy of an infectious disease, particularly that is widely transmission and transboundary or pandemic nature of the infectious disease. For agent, there are some factors need to consider, particularly the causal microorganisms, how its virulence is, how it comes and how it interacts with the susceptible host and how its genetic properties are. Then the host, particularly the susceptible host factors include also host genetics, host immunity, even individual or group is the demographic and that particularly contribute whether the disease will go bad or good. Environment also play an important role to make this disease severe or less severe, particularly the environmental factor, uh, the psychological, physical, and biological environment of the habitat. And over time also determines particularly the incubation period or latency period or length of disease process and times and trends of cycles. A disease first come in contact or get infected, and some infection or some microorganisms takes more time to make severe disease than other organisms. This is we called disease process. I mean, from getting infected to show the clinical illness. So some microorganisms, whatever the virus or bacteria, take a little bit um, longer time to make the clinical disease happen. But some microorganisms take very short time to make the patient so severely sick. For this particular SARS-CoV-2 cases, there are two sorts of particularly this causal agent also has rapid mutation characteristics. It's an RNA virus. It's a nasal, respiratory infection, and some other indirect cause that uh, directly involved with the viral transmission. All these things make the disease so dangerous. And particularly for this SARS-CoV-2, if we fix or compare this scenario, this pandemic scenario with the model, then for the agent, it is not fully understand now how the SARS-CoV-2 virus, how the characteristics of the virus, how the mutations is happening, because it has spontaneous mutations, and every moment is changing, and we don't know what is coming next. That is a bad side. And environment also changing over time. Human being, from the host side, our lifestyle changes, our belongings, our surroundings, our food habits, also our working styles. So many host factors are influencing to this disease happening. So from the host side, what should we do is we need to protect and we need to educate the particular patients when they get exposure. And from the patient side, we also need to educate about social distancing and quarantine when we interact with the environment, particularly the risk environment. And from the agent side and environmental interactions and agent, if we need to improve the sanitations and disinfection. Then, if we break the cycle from agent to environmental interactions, or environment to host interactions, or host to agent interactions, there is three tier. If we break down these three linkages, then it is possible to minimize the infection, or the EPICAR goes down, or at least keep flattened. That is the most desirable things what we are expecting nowadays. So epidemiological risk factors actually, uh, risk factor actually the indirect cause of disease. The direct cause is the particular microorganisms. In this case, is SARS-CoV-2, and the risk factors is other factors that is not the direct cause, but enhance the probability of getting infected, or probability or enhancing the transmission, or probability of getting severe diseases. All sorts of these indirect factors we called risk factors. Risk factors actually, here we divide it into two types. The factors particularly enhance or associated with the transmission and infections of this virus. If we can reduce or subside or diminish this kind of risk factors, then we can prevent the disease. That means we minimize the next infection. That is the if we avoid the direct or close contact, then we can avoid the infections, and we can break the transmission cycle. That is the way we can prevent the SARS-CoV-2 infection. And the other hand, this is actually associated with the control strategy. Control strategy we applied when it's already affected or the disease already happening. How can we minimize the loss? In this case, how can we minimize the death? That is the background of control strategy. The risk factors associated with the disease pathogenicity or severity of the disease. If we minimize these factors, that is the control strategy, that we can reduce the morbidity loss and mortality loss. But we cannot prevent the infection because the infection is already happening in these cases. So these two types of risk factors we consider for the prevention of next or subsequent infections, or the designing the control strategy for the patient who has already suffered or get infected. For this, risk factors associated with control strategies are age, sex, existing health problems, including asthma, diabetes, cardiac disorders, immunodeficiency. The patients get infected with SARS-CoV-2 and they already have in age groups, the elder age, or there is a sex issues also. I'm coming next. And the existing health problems, if the patient is suffering from previously suffering from respiratory problem, cardiac problem, diabetes, kidney problems, and particularly if he is suffering from immunodeficiency, that is a high chance of this patient uh, not survive. I mean, they will go to death. That is the risk factors we need to consider when the patients already got infected. How can we minimize the morbidity and mortality loss? It's a graph showing how age and sex are related with COVID severity. Most of the cases, the older peoples get uh, more. This is actually the gender. I mean, the female is the red color, and male got the it's a blue color or light blue. That means most of the cases, male has more severe infection comparatively to the female. And the comorbidity that we just talked before, that some other health problems, if already have it, if already he or she has, there is a high chance of getting severely infected and followed by death. Among them, this is a report we extracted from the WHO site. There is one case, case study analysis from the existing database. Hypertension and diabetes, the patient is 4.6% cases. Most of the cases, asthma is fatal, and particularly the hypertension. The patient who has hypertension before, as the case fatality rate is about more than 9%. And cancer, other sorts of malignancy, if he already has before, there is 10% fertility. Diabetes is more, 17, and the highest case fatality found in the patients who has cardiac disorders, particularly any sorts of cardiovascular disease, they are chances high. Environmental factors also, we talked it before, that many environmental factors, particularly the humidity, temperature, air transmissions, the information now comes is aerosol and airborne transmission, fomite transmission, all sorts of transmission. And this is another myth. Sometimes it causes there's many other coronaviruses transmitted through insect or some other arthropod, but in that case, no. This is no. And all these environmental factors influence the virus transmission to the from the infected patient to susceptible patient. So winter season, there is some sort of evidence that winter season has a higher risk of any sorts of respiratory infection due to humidity and temperature. In the hot humid weather, compared to the cold weather, particularly the respiratory infections get more severe, because what we call the risk factors for the respiratory infection, particular microorganisms is a direct cause. So many other risk factors are there, particularly winter is a stress, and in winter season every people usually get at least one times the natural or normal flu. This is due to due to seasonal stress. That's why cold weather is a stress to human body, and its coincidence or correlated with the SARS-CoV-2 infection. One thing is here, basic reproduction number. It's an also an epidemiological parameters and context. This is actually the indicator based on the epidemiological theorem. It's a numerical number, and this is the parameters based on which the assumption are made whether these viruses or this any sorts of microorganisms transmit to the susceptible host and particularly cause the infection. If a particular microorganism has basic reproduction number, we call this R naught. R naught is one or more than one, that makes or that can make an outbreak. And particularly if a R naught value is 0 or less than 1 or less than 1, usually it cannot make so severe infection, or its transmission is not that much high enough to cause severe pandemic situations. In this case, the SARS-CoV-2, the R naught value is nearly 2. That's why it has been causing severe outbreak, so rapid transmission and worldwide transmission. And this R naught value also considered when a vaccination schedule is designed to a particular population. If the R naught value is 1 or near to 1, then vaccination design is a little bit easier, and not whole the population should be under vaccination coverage. But on the other hand, if the R naught value is more and more, higher, then it needs much more coverage, more vaccination coverage to get the population immunized. That is called herd immunity. I'm just escape this slide. This is also an issue. This we already explained why winter can cause higher infection. Anyway, let's talk a bit about the impact of COVID-19, particularly in the agriculture, and even more in livestock, fisheries, and crop science, and also in other agricultural sectors. COVID actually had a great impact on action and activities of humanity all sorts of human activities are changes due to worldwide travel restriction and the strategic step-by-step lockdown. All sorts of free movement are restricted, particularly to reduce the human-to-human contact, the close contact, to break down the infection channel or transmission cycle. So agriculture is a part of integrated part of human activities, particularly the Asia Pacific region and particularly in the rural agro-based countries. So agriculture is not outside of this impact. Rationally, agriculture has also suffering a lot from this COVID pandemic. Two types actually. For the food, food is available due to transmission or travel restrictions. Food cannot reach to the consumers. This is one issue, and due to travel restrictions, people cannot move, and due to these movement restrictions, people are not spending enough time to produce food. So food is producing less, and food is getting available to the consumers also less. That is actually making more severe upcoming scarcity probably in the food. Anyway, still we are lucky enough that world what was speculating that maybe this COVID pandemic will create a serious food scarcity, particularly these developing countries. Still the situation is not that much worse. People are getting their daily foods, though there is a little bit bad scenario compared to other regular times. This is alarming, and with the agriculturists, particularly people who are engaged in crop production, animal production, fisheries, are should much more aware how should we keep continue animal productions and make their transport, particularly the food from production site to the consumer side, particularly what we call the farm to fork. This farm to fork value chain should be continue, and it should be a supreme priority to keep the people alive and fight against this COVID pandemic. This is actually a little bit back things that how we manage and pandemic outbreak. This is an theoretical things. Major steps are anticipations and predictions. Prediction is an important thing, particularly prediction science is now getting more attention after this pandemic happening. There are a lot of algorithms, epidemiological algorithms and computational approaches by which scientists can predict what is going to happen in next times for these pandemics, how it's going to down or up, or what is coming bad or good. And then there is a steps of preparedness. If the pandemic happens, then how should we fight off? What sorts of preparations we need to keep ready? Then another steps is early warning or surveillance. If there is very well warning or surveillance systems works, then and outbreak or pandemic outbreaks can be under control. Then effective and coordinated response. We predict what is going to happen, we get ready, or we have enough preparations, and we also keep the surveillance systems, and we got the early signals. Accordingly, we need to respond or take the action coordinately to manage the outbreak. And we should have a continuous evaluation or monitoring of the systems, whether all the systems is going well or not. And early case diagnosis is an important issue, particularly for the developing countries or densely populated country like Bangladesh, Indonesia, or many other Asian countries. People are not much aware, and infected people are getting in contact with the susceptible patients. People are not caring about the government request or instructions to avoid mass gathering or travel restrictions, or particularly the lockdown or keeping home. In such a scenario, early diagnosis, so early diagnosis is the best things and important things to minimize the bad consequences of a pandemic like COVID-19. So for this early diagnosis, diagnostic tools, particularly for this case, real-time PCR-based detection is recommended by WHO, but some other serodiagnostic tools are available based on antigen or antibody. Those antigen or antibody dependent test has some sort of limitations, particularly uh, the screening test. If the sensitivity is not that much high, then there is a chance of getting false positive test, or the specificity if goes down. If the low specificity test is used, then also there is a chance of false negative test. So this false positive test and false negative result is the limitation for these antigen and antibody based screening systems. However, if a highly specific test is keeping under regular screening, particularly for the pandemic situations, uh, because in this case, we need to make sure that no false positive patients are detected. Here is false positive is not that much worse if the patient we say is um, COVID positive, but really he is not positive. In that case, maybe he or she get a little bit depressions or something, but in reality he is not infected, he will definitely be cured. So that's why false positive is not that much bad things. Bad thing is that false negative test. If the patient is really infected but he tested negative, and what happens then, the patient says that's okay, I'm not COVID positive, so I can move around or whatsoever I can do, I can make a gathering whatsoever, he's free. But ultimately after 3 days he got severely affected and maybe he can die. And within this period he has spread the disease with so many patients. That's why false negative is the important concern for these purposes. We need to make sure that false negative patient is zero. That makes the transmission cycle break or keep the epic curve flattened. Particularly the serodiagnostic test in some cases, because the pandemic is already more than 1 year now. So we expect that there is herd level immunity is happening. So for evaluating the herd immunity, the serodiagnostic kit are sometimes useful, though having their limitation. A little bit about herd immunity. It's an phenomena that's particularly for an infectious agent. Body has the specific immunity to this particular antigen, and this immunity is a particularly the higher proportion of population that can resist the subsequent infections or exposure. And as a whole, community become exposure. It's a phenomenon that's, for example, 100 people in a community. Among them, 70 people get the immunity against SARS-CoV-2 by vaccination or by other natural infections. If the patient get natural infections but he survived, that he also get immune. Or another way is vaccinations. These two way, one person can be immunized. Either way, if the proportion of immunized people in a community is higher, close to 100%, is the herd immunity. The percentage, how many percentage or proportions of people need to be immunized to protect the further infection? This is a question. I mean, what is the threshold for herd immunity? There are a lot of debate, and the proportion or the percentage is much variable depending on the particular agent. But most of the cases is 60% or up is the threshold. If a particular community, 60% people get immunized with a particular agent, then the whole community will be protected. There is one report of this SARS-CoV-2 herd immunity, and they reported 43% populations, if they immunized against coronavirus infection, there is a chance of get protection from the subsequent infections of the community. So with these things, let's come to end of these presentations. I'm going to finish it in two or three slides. So here I'm talking about something, the immunity things, because it's a viral disease. Actually, any sorts of viral disease, there is no specific treatment. Bacterial disease we can prevent through antibiotic, even though nowadays antibiotic resistance is a major concern worldwide. But for viral diseases, no specific treatment. As a medicine people, we are saying like this, that viral disease has no specific treatment. So for viral disease, what we can do is prevention is the best things what we can do. But for SARS-CoV-2 infections, prevention is not working even. So what at least we can do is we can increase or improve our immune systems to fight against these viral diseases. Actually, viral disease, for all sorts of viral diseases, there I showed one slide that's host virus interactions. This is the equation actually. This is a fight between host and virus, host immunity and the virus particle. If the virus win, then disease will come, people will die, or severely bad things happen. Or if the host immune system is strong enough, then the virus will fail and the no disease will come. So how can we improve our immunity, immune systems? It's mainly particularly the healthy lifestyle is a key. We can do regular exercise. We can have a healthy diet, balanced diet, taking probiotic functional food. Probiotic functional food, particularly the yogurt and probiotic cheese and other fermented food is getting more popular, particularly the vegetables and other fermented food. So having fermented food in the routine diet is getting much more popular, particularly during this pandemic situation. It helps to improve our immune systems and fight against the pandemic, the COVID infection. And we need to have good and quality sleep. And we have to keep ourselves or our mind far away from any sorts of stress, physical, mental, any sorts of stress. We need to be stress free. That makes our body to cope up with the infections or upcoming infection. And the people who have bad habit like smoking, alcohol, or drugs, strictly prohibited and should be avoided now. So finally, this is maybe the final 2 slides actually. What we should do is it's a pandemic. Even though we should not be panicked, we should be careful. As it's a viral pandemic, we cannot deny, we cannot avoid, but we have to survive. We have to make ourselves strong enough, particularly what is said, our immunity need to make strong enough to fight against. So this is actually the key. We need to make our immunity up. This is some sort of things. We are also we all know that, so we need to keep physical distance, social distance, to avoid the virus transmission cycle or exposure. We need to improve our personal hygiene. We need to change our so many things actually. This is some sort of health rules and hygienic practices we need to adopt, but no need to be so worried about this. No need to be so panic. We should be careful, and we should try to maintain a healthy lifestyle. And finally, what I told before, that we need to pick the right information from the authentic source. Here some sort of sources. There may be an authentic information regarding SARS-CoV infections. So we need to pick the right information to make ourselves understanding really on the situation. So with these few words, actually, I want to say thank you all. So I wish you all stay safe and boost your immunity up. Thank you very much.
Okay, thank you very much, Professor Aminu. Such a very nice and interesting presentation. So we got a brief and clear explanation actually about the COVID-19, how we can avoid from disease, and how the world is still struggling to overcome this pandemic situation, especially with the emerging of the new variant of the disease. And we already have several questions from Amino. We already we wrote it's almost 9 questions, but we should we show it the question to the room, so we can, or we can let's say we can, because there's some question that are quite similar, Prof. Amino. And before we are going to the question from the chat room, there's Muhammad Zuhaib, I think he raised his hand. Okay, Muhammad Zuhaib, can you ask directly to Prof. Aminu? Time is yours, please. Mr. Muhammad Zuhaib. Okay, while waiting from question from Muhammad Zuhaib, there is already 9 questions, Prof. Aminu, because there's a very interesting topic, yeah. And I do apologize that we already what we call it, collect in the because it's quite similar, yeah. The first one is from Azhar Rai. Assalamualaikum, sir. He's from University of Muhammadiyah Malang, East Java. He asked about how the COVID-19 mutation or mutate so fast, how it can be. And in Indonesia, there has been a lot of many variation of this virus. I don't know if it's true or not. And the similar question is from Ahmad Bestari, I think. from IPB University. Assalamualaikum, and he asked about how much possibility the COVID-19 virus will continue to mutate, and are any study already estimated when the COVID outbreak will end, and why the COVID virus is so serious or massive in Asia countries compared to Europe or America. And then I think similar question. Perhaps the committee will try to show the question, yeah. Pak Jimas, Pak Arma can help us to. And then from Mita Sari, Mulawarman University, East Kalimantan. Does spraying people with disinfectant lower the spread of COVID-19? Because in my region, every day there's always spraying people with the disinfectant. And trends of COVID-19 incident in every country have different. Pardon, Prof. Amin?
No. It's okay.
Okay. Trend of COVID-19 incident is in every country have different depend on the region. Do you think which strategy we must to follow or make for country like Indonesia who has 1,000 more islands? I think it's a quite similar question related to how the virus mutation and how the strategy to avoid the virus itself. And after the answer from Prof. Aminu, perhaps Mr. Muhammad Juhaid can ask directly to Prof. Aminu. Please, time is short, Prof. Aminu, for answer the question.
Thank you very much. There are so many questions, but the things I got, the messages are more quite similar. Major concern is the mutation issues of these viruses. What I told that it's an RNA virus. All sorts of RNA virus usually get mutated spontaneously. And the same thing is happening for these SARS-CoV-2 viruses. And what we already have been observing that the different strains affecting in different regions, Brazil, and the latest one is India. The India strain is we call this Delta variants, and the mutation is still ongoing, and it's a prediction, speculations that it will continue so far. And how long it will continue, it's simply we should say we don't know. But what we can predict is that being an RNA viruses and getting the available hosts to survive, it could continue mutating. And the situations in Asia Pacific region like India, Bangladesh, and Indonesia, environmental conditions and the human density is more or less similar, particularly the recent bad experiences in India. Delhi and Mumbai is the highly dense city, and this is the virus actually. The transmission cycle is the direct transmission cycle is man to man. So where there is close communication, close contact, breathing, a transmission cycle is strong enough, and there is a high chance of getting infected and continue the infection so fastly and so rapidly within the community and the neighboring community, what they have the direct or indirect communication channel. And the country like Indonesia, though also a densely populated country, but the Indonesia has a different characteristics, different geographical characteristics. It's an island country. There is a natural advantage actually. It's a things of positivity and hope because of this island. It can be easily separated from other countries. Those are having endemic or now is pandemic and particularly if you say pandemic with this very dangerous strains, Delta or other Lambda variant also is coming. So there is a good thing for Indonesia and other island countries like Japan or Australia, New Zealand. If they close the air border, they can easily transmit, easily break the transmission cycle. But question is on the other way around, because of international connectivity, human life is so usual to international communications and roaming around the globe. Lifestyles and living standard, business, and every sorts of life event are not fully independent in a particular country, like a low or middle income one. However, Indonesia has this natural advantage of having the surrounded waterland, particularly because of island. They can be isolated from other countries if they restrict the air border. For example, if I say from our recent experience, I mean Bangladeshi experiences, people are suffering from the latest SARS-CoV variant and the Delta variant. Few weeks ago, actually, India had been suffering a lot with this variant, and Bangladesh has at least three side land border with India, and almost surrounded by the India, and so many communications people are used to have with India because of business, lifestyles, or so many others issues are there. Treatment, so many other issues are there to communicate or travel between India and Bangladesh. So the travel restriction was there in written, but in practical there was not that much. And result is actually the bad. What we are last 5 or 7 days is getting very higher number of death and by higher number of new infections in Bangladesh. And the variant we reported here from our national level laboratory, they reported that at least more than 65% affecting the Bangladeshi people affecting, those are having the Delta variant that was reported in India nowadays and in the recent past time. So this is the bad things. If we have the land border from a highly infected area, there is a less chance to escape. But if we have the natural advantage what Indonesia, Japan, Australia, New Zealand have, the water surroundings island, they have at least a chance to escape themselves from the direct land communications like India, Bangladesh has. They share the land border. However, things actually what is going to be next and how long the pandemic will continue and how bad the things will come. These sorts of things are kind of speculations. Nothing we can predict rightly. But from my side, what I would say is that simply get ready for the worst. If something good happen or something at least better is coming, so we are happy. But we need to get ready that the worst thing is coming. We need to get prepared, or we need to design our protective stuffs at least from the individual level to national levels to prepare for the bad things is coming. So we cannot control, and not everything is in our hand. What the individual we can do is keep ourselves avoid the exposure, avoid the mass communication, particularly the direct transmission cycle, avoid the direct transmission route, transmission cycle. Though it's how long we can keep ourselves isolated. This is a big question, and life should continue, and work should also continue. Work-life balance should also be made. So many other things are coming. People are getting bored, so everything is going on, but life is first, and then other things. So individual level, from people, from the individual level, what we can do, keep ourselves, at least keep myself protected. This is the individual phenomena should be like. And from the country like developing world, also a big issues for the food. uh, Government has declared for the lockdowns to stay home. The poor people, they need to manage their daily food. They have to go for work for managing their food. So if the scenario is like that, how can government restrict the people to stay home? That is the burning issue what the developing countries is facing with. Uh, Actually, these are the things. All the questions and discussions. What we are doing is that has no particular specific answer, but there is a concern. There is a concern. We need to discuss, and we need to get ready and get prepared accordingly.
Professor Aminu. There is another question, but there will be directly by the participant. First is Muhammad Zuhair, and the next Miss Ellen Naisinto. I think the first one is Muhammad Zuhair, yeah? And then continue followed by Miss Ellen Naisinto. Please, Muhammad Zuhair. And afterward, there is still a question in the chat room that we can read together. Okay, Muhammad Zuhair, please.
Muhammad Zuhair, are you there, please?
Or is not yet ready, please, Miss Ellen May, to ask the question directly to Prof. Aminu. Time shares.
Okay. Good afternoon, everyone. Am I audible?
Yes, you are.
Okay. Hi, sir. I'm Ellen from the Philippines, and I would like to ask: is it possible that the coronavirus transmission? I'm sorry. Can you hear me?
I can hear. Please proceed.
Okay. So, sir, is it possible that the coronavirus transmission went faster if it is influenced by the country's weather? Like, for example, here in the Philippines, it is very hot because we are in a tropical region. And is there a possibility for that the coronavirus spread too fast than like the other countries that it is cold, like in the weather? Is it?
You are talking about the environmental conditions, whether environmental conditions influence the transmission become faster or lower, right?
Yes, exactly.
Take it rightly. Thank you. Thank you for the nice question. Actually, these are the common things and issues we have been talking about early months of last year. That particularly the Asian regions got a lower number of infection cases compared to the cold countries like UK, Germany, Italy, and some other and USA. What they have been suffering in the early stage of pandemic, so huge number of new cases. In the other hand, in Asian countries like Indonesia, Bangladesh, India, Pakistan, Philippines, Japan, getting lower number of infection. That is actually not clear, but what other respiratory infection is happening, that cold environment, but cold weather has an influence for virus survivability and also virus transmission. If the virus is an airborne infection, that is the latest information is coming. then there is a chance of getting rapid transmission uh, for the warm, hot, humid weather and heavy wind flow, particularly the monsoon weather. In the monsoon weather in the spring and early summer, what we have, there is a huge and higher wind flow, and it has an influence to the transmission cycle or speed of transmission enhancing. I think even but the information actually it's not crystal clear enough to say like a yes no questions or just a absolutely say whether the uh, environmental or ambient temperature rightly influence the COVID transmissions rapid or slow. This is actually a dilemma the scientists are facing with actually. There is no, I would say there is no crystal clear yes no answer actually with this. This is still not clear enough to say the absolute answer. Thank you.
Okay, thank you, Professor Aminu. I do hope the answers quite satisfied for Mr. Alumni. Yeah. Okay, next from Muhammad Zahid. Are you there? Okay, if there is still not a response from Muhammad Zuhaib, there is another question in the chat room that we already compiled for Prof. Aminu because it's quite similar. And actually, I think already explained, but perhaps you can make it more briefly. From Shafira, IPB University, Putri Sausan, IPB University, from Mr. Nahiduzaman, Bangladesh, and Muhammad Mustafizur Rahman. They asked about, is it possible the COVID-19 infect to the animal or livestock, vice versa, from animal or livestock to human, and human to the livestock or to the pet? Please, time share, Prof. Aminu.
Okay, thank you. It's also an interesting question that has been raised even from the early pandemic days, whether this SARS-CoV-2 has a genetic potential or not. I mean, the genetic disease we call or infection we call, the microorganisms can be transmissible from human to animal or animal to human. What I have shown in the slide that coronavirus is, this one is not new actually. It's a new variant. Coronavirus exists from very early or very beginning. And some strains have been affecting poultry, some strains have been affecting animal. The question is whether the same strain infect to human can be transmitted or can be separately infected to animal or vice versa. For the novel coronavirus, the SARS-CoV-2, actually the answer is no. It has no genetic importance. But this is the issues. But some report actually from Thailand, Belgium, USA, and some few other countries, they have reported that SARS-CoV-2, they can isolate it, the SARS-CoV-2, even the genomic analysis they reported the later on. They reported that SARS-CoV-2 virus have been identified from the zoo animal, particularly the tiger, and some pet animal, cat, and some other hard object they have been identified. So this report later on could not be directly interconnected, or they cannot be traceable whether this infection comes directly from human being. The isolated organism has similarities with the human isolates of SARS-CoV-2, but the question is that it was not clearly evident that this SARS-CoV-2 isolated from the animal species are directly transmitted from any source of human patient. That was not clear yet. With these informations, we cannot say that SARS-CoV-2 is transmissible between human and animal. And this is until now the informations. But it is reported that the same thing, same virus is isolated from human and animal. But the issue is the no clear evidence of this transmission cycle between human and animal and vice versa. Thank you.
Thank you, Prof. Aminu, for the answer. Okay, the last question, I think. I do apologize if there is still question that not yet answered. Perhaps the committee will send the question directly to Prof. Aminu, and then the Prof. Aminu will give a response for that by email or something. Okay, the last question will be from Puspita from IPB University. She said that last time I was infected, Prof. Aminu. I was 19 years old. I always obey the health protocol, staying at home, consuming healthy food, and doing some exercise 3 times a week. But I get infected. In contrast, my friends who ignore the health protocol and always go outside are not infected by COVID-19. Could you tell us, or could you tell me why this case happened to me? That's the last question of this session, Prof. Aminu.
Thank you very much. It's a very practical question actually, and really interesting question. Actually, let me say an example. When two friends get all day long wet in the natural swimming or raining, but one day after, one patient got natural flu or a little bit fever, but another friends are still fine. So what could happen behind? They got exposed with the same microorganisms, but one who got infected, even though it's not SARS-CoV, it's just a fever or cold even or flu even, what happened with his, he might be not immunologically strong enough to resist his early infections or any sorts of stress like swimming or long time waiting under rain. And the other hand, the friends who keep fine, his immune system is strong enough to fight against the exposure of raining, swimming, or any sorts of infections. The same thing happened for SARS-CoV-2 also. The key is the host immune system, whether the host immune systems can strong enough to fight off the exposure or exposed SARS-CoV-2 organisms, or the host immune systems got failed to fight off. If the host immune system get fight or unable to fight off, he got infected. If he survived, that means he are strong enough to fight off. And another thing is also an genetic background. Particularly, there is individual variations even our human being and also all sorts of livestock species. Individuals' genetic architecture has contributed a lot to influence the strength of immunity. If the person has a strong genetic background, this is not strong actually, this is a genetic state. We studied is a perspective of immunogenomics. The person's genetic architectures, those are closely related to immune system function. And if the genetic parameters or particular parts of genetics, and those has high immunity or immune system control, the person, and even though they or he got exposure, but they didn't get infected. On the other way around would be happen. That's why the persons who are getting exposure, who are regular coming outside or doing everything, get exposure, coming contact with susceptible patients or coming contact with corona isolation center, even the infected patient, but not really get the infected. That's the issue his immune system has, and his genetics and his individual strength to fight up this coronavirus disease agent. On the other hand, you might not have this criteria, particularly your immune systems, your genetics, and your individual strength may not strong enough to fight against. Even though you are not getting outside, you are not getting exposed to other infected patients, somehow or a little amount. What I said, there's the dose of infection also an important issue. Some people get lower dose but get infected, but some people exposed with higher doses of virus but didn't get infected. This is the issue. What in your case happened, it might have that your immunity, genetics, individual is a little bit relatively lower, and you might have got exposure with a higher number of infectious agent by any means, though you are not sure that how you get the infection. You probably not go outside, or there is a little chance of getting exposure, but it's sure there was a chance. You might not aware of how you got infected, but it is sure that there was a chance of infection, there is a chance of exposure. You might not notice particularly on which way of transmission or exposure you got it, but the things is that your infection, your immune system, genetics, and individual was not strong enough to fight off. I think this is the issues we are talking about. Thank you very much.
Okay, thank you, Prof. Aminul. Again, we do apologize because the time is up. Probably for some participant or they still have a question, perhaps again, you can ask directly to the committee, and the committee will send the question to the Prof. Aminul, and and we hope that Prof. Aminul will give a response for a question that you are not already addressed to Prof. Aminul. Again, Prof. Aminul, thank you very much from us. And before we end the session, there will be a certificate appreciation that will be delivered by Professor Asep Gunawan. Professor Asep Gunawan.
Yeah, Pak Edit.
Okay, I will share the screen, yeah, Professor.
Okay,
For the
thank you,
certificate
Pak
of
Edit.
appreciation.
Yeah.
Okay, this is the.
We are really appreciate for you already giving the lecture. Otherwise, I know you are in not a good condition. We are pray for you become better soon and health again, Duta Besarung. So we would like to thank you for giving this lecture. So on behalf organizing committee, so we send e-certificate for you. Yeah, we hope we can look forward and further collaboration in the future. Thank you.
Thank you, Dr. Asep Gunawan, one of my very good friends, actually. And really, what I would like to say, it's a great honor for me to be here. And for the participants, I'd request if you have any further related questions to me, I'm available through email. Actually, I'm shared with. So I would be happy to communicate with you next. And for the organizer, I must acknowledge you and thank you very much for giving me the honor and the certificate of appreciations. I would like to continue with the IPP University for further academic and research actions. And finally, thank you very much. Thank you all. I wish you all the very best. Keep safe and stay fine. Thank you all the participants and the organizer. And finally, thank you. Thank you very much.
Okay, thank you very much for Aminul. And then before we give to the committee, perhaps we can take a picture together.
Yes,
Please,
yes.
Pak Jimas and Pak Ferika can help us to take a picture. Yeah, there is a seven slide, I think, or 7 group. Okay,
better
And
to
give-
have the video on.
Please turn on the video actually, so we can have a video. We have to take a picture, Prof. Aminu. So perhaps 2 minutes before you will leave. Okay, Pak Arman. Yes. Okay, slide 1. Okay, next slide 2. Yes. Okay, thank you, committee. And then after, thank you very much again, Prof. Aminu, Pak Asep. And then we will meet again in the next, in the second session. And I will get back to the committee for the next program before the second session will begin. Okay, again, Prof. Aminu, thank you so much.
Thank you very
Stay
much.
fit and healthy. Pak Asep also. Please, committee, because after this, there is some video, yeah? Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you very much.
Waalaikumsalam.
I'm leaving now. Thank you very much.
Okay, you're welcome.
Thank you.
Have you heard this story about a beautiful place? A place that makes you feel restful. In the midst of people who live in religious harmony. A place that makes you feel home, that makes you curious for more. Adventure. Start your journey. Travel to the place where the story unfolds. Feel the sea breeze caress your face and the sound of the waves breaking your consciousness. That you can see them through the beauty of woven fabrics.
Yes, Prof.
Or smell them through the scent of coffee that wafts in the air. If you have never heard it before, you may now find it intriguing. You may want to know every square inch of this place. From the beauty of its land to the depths of its seas. With an amazing biodiversity. You've heard this story about a land which has traditions so diverse. Meet these friendly people who will welcome you with warmth. After all, welcoming guests is their tradition. The light of Aceh. Wonderful Indonesia.
Very nice video.
In the legendary landscape of the largest crater lake in the world thrives a glorious civilization. In North Sumatra, people live colorful lives. And nature continues to fascinate both the locals and visitors from all walks of life. A delightful escape with a sense of adventure. In the hidden paradise of Sumatra, experience the awesome nature. Understand the connection to ancient cultures, like dancing for ancestors, a colorful tradition, joyful yet also potent, mystifying rituals, proud people of the mountains. At the coast and in the cities. Remarkable diversity. A bustling city. A multicultural Mecca. Peaceful harmony. Incredible culinary. Astonishing wood carving. This is North Sumatra. The land, also the people. Friendly faces and genuine warmth. Wonderful Indonesia.
West Sumatra, famous for its ancient history, echoing through its canyons. Wonderful contours formed by the tectonic slabs, with fertile volcanic land crossed by the grandeur of Bukit Barisan. Ageless natural beauty is felt every step we walk and every sight we see, leaving behind countless tales of myths, hard to be neglected. Catch the beauty of Lake Maninjau, wonderful landscape is witnessed when we fly over the top of Lawang Hill. Priceless high elevated adventure. Pedaling together around Singkarak Lake. Experience the phenomenon of walking on the living bridge, while the waterfall refreshes the travelers. Evolving with time, without letting go of the inscribed history. Villagers keeping their joyous traditions. Pachujawi, which means cow race. The elegance of martial art. Silat. Visiting the Pagariun Palace. Acknowledge the culture history of Minangkabau. The enchanting plate dance will greet guests, exposing their striking physical and vulnerability. Taste the various traditional culinary of West Sumatra. Padang main dishes uniquely served to your table, well known for their savory and spicy flavors. While cakes and beverages are exceptional. The spicy balado cassava chips. And of course, the legendary delicious satay padang. Delight in the beauty of silver crafts in an elegant ivory nuance. Try on the rich, colorful sun-kit fabrics and carefully attempt to knit in this house of weaving. A new experience you can bring home. Take steps through a history. From Japan's bunker to the biggest coal mining in its era. Encounter the splendor of West Sumatra and merge into its glorious beauty. With the power of history flawlessly intertwined with the wonders of nature. Wonderful Indonesia.
Okay, the committee, the video is it over yet for the video? Pak Jimas, Pak Arman. Okay. Okay, thank you, committee. And then welcome back, everybody, to second sessions of our online summer course program at Department of Animal Production and Technology. I do hope a little break can make you still awake, yeah? Still enthusiastic after a cup of coffee or tea, so you're already refresh for the next session. Okay. Did Prof. Asep already join with us, Prof. Asep? Hello, Prof. Asep. Still with me, Editor Aditya, for the host of this second session, yeah?
Hello,
And,
Prof. Adit.
Okay, Prof. Asep, thank you very much for already join, yeah? Okay, the second session will be presented by Professor Asep Gunawan. And before we start again, we are from the committee kindly remind you to still turn on the video and then from until the end, yeah, turn on the video but mute your audio except for the discussion session or the committee asking you to unmute your audio. So please, you don't have to put a static video or something during the lectures. If there is some error or the problem in the connection, you can turn off the video. It's better, I think, compared to you put a static video or something of yourself, yeah, or a portrait of yourself. I think it's better if there is something trouble with the connection. Okay, and then you can ask the question directly. Raise your hand, and then the committee will help the participant to ask after the presentation, or you can write the the question on the chat room, yeah, and me as a moderator and also the committee will try to compile the question if there's a similar question, yeah. And before we start, let me explain a short CV of our honorable lecturers for this second session. Where is it? Yeah. Okay. Can you see? Can you see the slide for the participant and for Prof. Asep? Can the participant see the PowerPoint? Okay, clearly, yeah. Okay. Our honorable lecturers for the second session is Professor Dr. Agrar Asep Gunawan from the Department of Animal Production and Technology. Prof. Asep is one of the youngest professor in IPB University. Yeah, and then he held a Bachelor of Science from Faculty of Animal Science, and then and hold Master and PhD program from similar university in Bonn. I think it's similar alumni, yeah, Prof. Asep with Professor Amino. Okay, and then so very productive in terms of publication. There's a lot of publication that has been published by Professor Asep Gunawan and his colleague, yeah, regarding to his field expertise in terms of genetics. And there's almost 80 publication, yeah, journals and also national accreditation. So if some students, especially abroad, would like to continue master or PhD program in our university in terms of the genetic field, livestock, so you can directly make a communication, a contact with Prof. Asep Gunawan. That's okay, yeah, Prof. Asep. There is some student who asking you for discussion later on about this, about your field of your expertise. And then Prof. Asep also not just on the write publications, and also become editors and reviewer in several journals, national and international journals. There's PLOS ONE, and then Animal Bioscience, Gain Report, and then Tropical Animal Science Journal, Indonesian Tropical Agriculture, and then Ilmu Pengetahuan, and also Journal Veterinaries. It's a very reputable journals, yeah. And also Prof. Asep already have a patent, Prof. Asep and his colleague, actually, yeah. The first one is leptin receptor gene for selection, fatty acid composition, and cholesterol in sheep, and also how to improve sheep meat flavor and others using candidate gain, CYP2A6, yeah. There's a second patent of from Asep, Prof. Asep, and then there's a short CV of our honorable lecturers this afternoon. And Prof. Asep, there will be a maximum an hours for a presentation, and followed by the discussion with the participant. Yeah. So, do you prefer for us to help you for the presentation, or you will be done by yourself, Prof. Asep?
Yeah, I will try by myself.
Okay, thank you, Prof. Asep. And again, participant, please. I think this is a very nice topic, yeah, that can improve your knowledge and also your insight, yeah, about genetic or biomolecular genetics. So again, please enjoy the presentation, and also don't forget to deliver the question to Prof. Asep. Prof. Asep, times and this Zoom rooms is yours. Thank you.
Okay, thank you, Pak Edit, for wonderful introduction, and then all participants, greeting from Bogor. So in this opportunity, I would like to share, some more knowledge, about, maybe this is a more specific topic, but I think maybe because this topic is also as a tool, that you can use for other interdisciplinary science, not only animal. Actually, you can apply in plant, in fish, in microbiology, maybe so. In this opportunity, I would like to a little bit share knowledge about that. Okay, so this is more related with the genetic background, so in this lecture, we will try to grip you knowledge about the transcriptome analysis in livestock. We focus on the application of RNA sequencing, for their application. So after we discuss, in the first lecture related with the current and update issue, related with the COVID-19, so maybe this case also can be included in our topic. So the outline of my presentation is consists, we talk about what is the transcriptome definition and the scope, and then also we talk about the RNA sequencing for introduction, and then how maybe if you want to perform or interest to conduct this kind of research, maybe you can see what is step by step, and then also we explain about the experimental method, and also how to analyze. This is the important, since yesterday we talked about the data analysis, how to handle. Maybe this is also important for us. And then also what is the summary after we analyze, what kind of data we can interpret and we can write in the paper, or maybe in regular report. So maybe this is important for us also. And then also the application of this kind of research, especially for research study. And also finally we conclude by conclusion. So I searched actually maybe few hours before, since we talk about the COVID-19, maybe no people know before, about the qPCR, qPCR, maybe only people who dealing with the molecular biology maybe know about the PCR. And also now people go more deeply again, not only PCR, but people already understand about the CT. What is the CT? If people, for example, has a COVID-19, and then people say, how about the CT? After like Isoman, after the quarantine or something like that, whether the CT is already decreased, or maybe it's already increased. So now people more understanding, not only in the genetic background, but also people who not really has this kind of background, like a social, maybe economy or something like that. People now is already understand. So like this also, how the transcriptome characteristic, this such a case of the COVID-19. For example, in this case, maybe this is already published 2021. This is very easy to understand. We know it's COVID-19 vaccine. Maybe they have blood. Maybe this is related actually with Professor Aminul. And then we such this a normal, and also this maybe they have maybe a vaccine with the COVID-19. And then people try to extract, the extract can be from the nose, or maybe from the blood before, maybe normally for the swab PCR from the nose, and then after that, and then they collect the sample from the nose, and then they extract. After they extract, and then they do PCR, yeah, and then from the PCR they can determine positive or negative like that. Yeah, so the same also when we compare between two different conditions, people who has normal condition, and then the one is vaccine for COVID-19, and then they can see what is the difference. Yeah, what is the difference between normal and people who has COVID-19. Yeah, so from this is the illustration. This is the control as a normal, and then this is the COVID. So they have a difference. Yeah, they're different. But the different we can have a look inside due to several factors. Like in this case, we talk about the genetic. Yeah. So people know the source of the COVID-19 is SARS-CoV-2. Yeah. But not only SARS-CoV-2, yeah, people now they try to find also several gene, yeah, several candidate gene who make differentiate between normal and also the COVID-19. Yeah, so this, for example, they found, yeah, EL6, yeah, EL6 or interleukin, interleukin 6 receptor and TPV3. So this is already published, and then they said maybe who make differentiate by this candidate gene. So maybe they have a need further more work, yeah, to characterize this gene, and then maybe after that, then they can produce, yeah, some such kind of the vaccine or maybe drug who can eliminate or maybe can solve, yeah, this kind of different like that. Yeah. Okay. So, and then we can talk about the SARS-CoV, yeah, maybe, and then if we talk about the animal, yeah, like a milk production, like a meat quality, yeah, like egg quality, yeah. So this is the complex trait, yeah, complex trait. Why is complex? Because this trait can be influenced many other factors, not only by genetic but also by environment factor. Yeah, and inside of the genetic also not only single gene, yeah, single gene, but has they several gene who control this kind of the complex trait. Yeah, complex trait like COVID-19. When we talk about the animal production, yeah, meat quality, yeah, milk production, yeah, and something like that. So people try to dissect, yeah, to separate, yeah. People try to know what is the behind of this story. Why, for example, people this has a suffer with the COVID-19? Why this may be a normal or maybe a little bit resistant? So people try to dissect, to characterize, yeah. So maybe they have some kind of different, yeah, in term of gene, yeah, in term of gene, yeah, or gen, yeah, maybe in Indonesia we call gen, yeah, bukan gene, yeah, genes, yeah. And also people try to characterize, yeah, in whole body, and then they try to map, yeah, to map the in each chromosome, yeah. Since our body is consists the total of the genome, and then inside of the genome they have a chromosome, yeah. So inside of the chromosome they try to put or to find, yeah, in which location, in which number chromosome. So people try to characterize with quantitative trait loci, and then people try to combine, yeah, to combine between the physical map, yeah, and also with candidate gene. So in this case, people try to characterize, yeah, with the genome-wide association study. Yeah, this is the illustration. This is the chromosome in whole body, and then they try to find in term of the chromosome, maybe they have several extreme, yeah, candidate gene who maybe affect to the phenotype, to the trait. yeah, to the thread, yeah. And also other, yeah, people can characterize using a tools, yeah, that we call as a transcriptome, yeah, transcriptome by characterized, for example, in this case is next generation sequencing, now is more familiar. So what is the transcriptome actually? Transcriptome is the complete set of the transcript in a cell, yeah, and their quantity for a specific developmental stage or physical condition, yeah. Like I mentioned before in the first slide, people try to know to quantify the cell, yeah, between the normal and the COVID, yeah, patient with the COVID-19. What this is the different development stage condition between normal and COVID-19, or maybe between normal and cancer, for example, yeah, between high milk production and low milk production. This is normally we found in our phenomenon, yeah. So the transcriptome, why we need to understand, yeah, because by transcriptome analysis is important for interpreting the functional element of the genome. Yeah, so we can know the element, and then we can have now, yeah, the molecular constituent. What is the constituent in the cell? Yeah, like before we found interleukin 6. Yeah, before people didn't know. Yeah, why is different between COVID-19 and normal people? And then understanding the differential express gene between two conditions. Since the body is the same, yeah, me and for example, Pak Edit as a moderator, in our whole body, they have the same, yeah, the same structure. Yeah, for example, we have the same number chromosome, we have also the same gene, but inside of the gene, since we are different, yeah, for example, me and Pak Edit, yeah, maybe in body weight, Pak Edit more higher compare me. So in such kind of the gene, maybe the expression is different. Yeah, the expression is different. So in this transcriptome, we want to understand how the differentially expressed between two conditions. So transcriptome, and also they have transcriptomic, yeah, omic and also ohm. Yeah, transcriptomic is study, yeah, when we talk about the genomic, yeah, proteomic, yeah, transcriptomic, and then our mind, oh, this is the study. What kind of study? Study of RNA in any of form, yeah. So maybe we have to remind again the central dogma of molecular biology, yeah, related with the synthesis protein, yeah. So the transcriptome is the set of RNA molecule, yeah, including messenger RNA, yeah, ribosome RNA, transfer RNA, or non-coding RNA, yeah, produced in one or population of the cell. Yeah, for example, you want to quantify the mRNA in the liver, yeah, in the muscle, yeah, in the brain, yeah. So this is the cell, yeah, in the cell. So the term can be applied to the total of the transcript in a given organism, yeah, to the specific subset transcript present in particular cell. So we want to know, yeah, in this cell. Yeah, and then this is to reminder again, the central dogma of molecular biology. You know, the first from the DNA, yeah, since the whole body, yeah, we can collect, yeah, the sample in DNA, and then from DNA, not all, yeah, DNA can be transcribed, yeah, maybe only in some part. Yeah, so we call as a RNA. Yeah, in this RNA, they have a process that we call transcription. Yeah, so from the transcription, yeah, and then we has the product of mRNA. Yeah, and then it will be continue, yeah, from mRNA to the protein. Yeah, that the process that we call as a translation. Yeah, and then we can see what is the phenotype. Yeah, so this is more detail, yeah, about the genome, the whole body, in the whole body, and then inside of the whole body they have a chromosome. Inside of the chromosome they have a locus. Yeah, in the locus they have a gene. Yeah, so the gene, and then they from the genome, and then they will transcribe to the mRNA, and then to the protein, this what can happen in the genome, and then what appear to be happen, and then this is transcriptome, and then what it make it happen, proteome, and then phenotype, so that we see, for example, what is maybe in animal, we have to take example in animal. Why is one animal could be high milk production and then one is low milk production? This is the phenotype. But inside of the different, otherwise we do the same treatment, the same feeding, the same environment. Maybe we have to look inside of the genetic. Maybe they could be different inside of the genome, due to the mutation, for example, or maybe the transcriptome, the quantity or the quantity of mRNA transcript, and also maybe could be in the protein, like that. Okay, so the transcriptome scope, unlike the genome, the transcriptome can vary external environment condition. Maybe the difference could be due to the environment condition. If you give a feeding different, of course, maybe it will be give the result different for the animal. So the study of transcriptome refer also the expression profiling, examine the expression of the level mRNA in cell population. So normally, because this is very small, and then they use high throughput technique, because we only quantified small particle, and then of course we have to use the high throughput technology. So what kind of technology that common to analyze or to conduct research for mRNA? They have a tool, in this case they have a hybridization based approach, normally common for this hybridization using fluorescent, microarray technique, and then the second is using a sequence based approach, that we familiar before, like a Sanger sequencing, and also now is common using RNA sequencing, like that. So this is the development, how the development people using the high throughput technique, since 1992, if we compare until now, maybe this until 2017, maybe we have left 4 years. Of course, here maybe they have a lot of technique, but you can see how it is very fast. People introduced in 1992, and then little bit longer, and then after that 9 year again, 9 year more, and then they have other technique, and then 6 months, and then 2, 3, 2 almost, he's more rapid, how they introduce, and then they get more technique inside high throughput. For example, this transcriptome sequencing they introduced 2009, and then after that, this is very fast, and then a lot of technique how to analyze the transcriptome. So this is the microarray technique. The same, the simple thing, we only compare 2 different phenotypes. For example, here they have a normal people, and then here is not a tumor, or maybe like a cancer, and then they label, and then they has like inside of the chip, they have a gene inside, and then we can blast, like what is scanning, maybe, like a scanning, and then we can see what is the difference between normal and tumor, like that, and then we can get the product in the tumor section, or in the tumor sample. They has gene which is high expressed, for example, but in the normal, for example, they have lower expressed. So this is can make a difference. So microarray, like I mentioned before, and then we can see what is the difference. Yeah, how about the microarray technique? Yeah, like now maybe people more familiar with the isolate RNA, yeah, a sample from nose and the same. Yeah, like for example here we collect from the tissue sample. Yeah, maybe it could be liver, it could be muscle, and then they try to isolate the RNA. Yeah, after that they convert from mRNA to cDNA. Yeah, this, and then we have two different, for example, sample. Yeah, and then we can see, yeah, what is the difference, yeah, between two conditions, which one is for example soul, and then which one is silent. Yeah, so this is the summary of the technique sample, and then they have a RNA extract, and then label, and then they hybrid, and then scan, and then you can get like a image, yeah, like that, like a photocopy, yeah, like that. So the thing is, after we extraction RNA and then scanning, how to handle the data, yeah, how to handle, because they have a huge number of information genetic inside of that. They have a lot of differential expression, and then they try to handle using the normalization, and then they calculate what is the difference between two conditions, normal and tumor, for example. Yeah. So how to measure it? To measure it, that we can say differential express, yeah, to see or to compare between two conditions, between normal and tumor, between normal and COVID-19 patient, for example, like that. So the difference could be, say, if they have, for example, different 1.5 time, yeah, 1.5 time. So to measure it, they have a way to score it that we call as a Paul chain, and also they have a statistic value, yeah, between two conditions should be p-value 0.05 minimum. Yeah, so how to calculate the Paul chain? Paul chain, we can calculate from two different conditions. The expression value, sample one, for example, COVID, and then we compare with the expression value by the sample two. Yeah, like now the city value. How about the city value between the normal, for example, the normal, for example, 30, 31, for example, and then we compare with the city value with the COVID 20, for example. Of course, maybe they have different 1 point something like. So this is how to determine the difference, yeah, between expression, or maybe the differential expression, two condition. Yeah. So this is the technique like I mentioned before, and then they can see, yeah, which the sample they can show, and then which sample they can not show. Yeah. Okay, so this is still related with the COVID, yeah, people from the COVID, this, and then they try to compare, yeah, transcriptomic, not only because they also people, they have other effect, yeah, like who people has a schizophrenia, and also post stress development, like that, and then also the people who have a bipolar, and then they have similar, yeah, they have a similar what is finding constituent element, yeah, like what kind of gene that can be maybe the same, yeah, in these three cases. Yeah, so that's why we have to reduce, yeah, the stress, and then good quality of life, yeah, good consumption of the high food quality. So because this is also related, yeah, maybe now every day we hear ambulance, yeah, and after that, and then sometimes they can decrease, yeah, our immunity or something like that. And then, yeah, this is correct, yeah, people try to make a correlation between the situation now, and then with other factor, yeah, with other factor or other activity, like that. Yeah. So what now they have two technology who can quantify or analyze the transcriptome. First is microarray, and then the second is RNA sequencing. So RNA sequencing is method measuring gene expression using deep sequencing. Yeah, like we say before. So the typical of RNA experiment aim to find differentially expressed gene between two conditions. Yeah, again, two conditions. RNA-seq can also be used to discover new transcript, yeah, like a splice variant and fusion gene. Yeah, so this is very easy. When you talk about the transcriptome, of course, inside of the information, they compare two conditions, yeah, and then from two conditions, we characterize what is the cause, yeah, to make a difference between two conditions. Yeah, so the technique of the RNA sequencing like this is the illustration. This is more better before from the Sanger sequencing. The RNA from the extraction of RNA, yeah, and then they convert to cDNA, yeah, like that. And then after that, they have like a here adapter, yeah, they put in the sample, and then they short sequence read of time. They alignment to the reference genome, yeah, to the reference. For example, if we are human, and then will be alignment to the Homo sapien sequence. If, for example, a chicken will be aligned to the Gallus, yeah, or maybe other, yeah, like a cattle, maybe they have a Bos taurus, yeah, they have already, yeah, in database, and then try to alignment, and then they can get the 3 result at least, yeah, exon. This is normally the number of transcript, yeah, and then also junction and also poly A and read, and then finally we can quantify the number of transcript. So RNA-seq is more, comprehensive approach, yeah, in molecular technique, because they can identify, yeah, several phenomena. Like SNP, normally they can analyze by genome, but in this case they can analyze also. And then small RNA can identify epigenetic event. This is more related with the environment factor, and also alternative splicing, and also in this our scope lecture, we can analyze the transcriptome. So how to design RNA experiment? Yeah, before we start the RNA experiment, some question that we should ask ourselves. Yeah, we want to analyze RNA. Yeah, but what do you want to compare it? Yeah, so that's why you have to list at least this question. Why do we expect to find differential expressed gene in the particular tissue? Yeah, now it's clear. For example, people now try to find the very good vaccine, yeah, very good drug, yeah. So that's why they try to compare between the normal, or maybe now the variant Delta, yeah, variant Delta, and then compare with the normal. Yeah, what is the difference? Why is variant Delta we have to use 2 masks? Yeah, but before 1 mask is enough. Yeah, so it means they more stronger. Yeah, so we have to find, yeah, we have to find more vaccine like that. Yeah, for the application next. Yeah, next. What type gene do you expect to find differential express? Yeah, at least when you talk about the SARS, the COVID-19, maybe they have the family gene related with that. Yeah, for example, SARS-CoV-2, SARS-CoV-3, SARS-CoV-19, if they are new or novel. And then what are the sorts of variability, yeah, for your sample? And what do we expect, yeah, most your variation to come from? So at least, yeah, that you want to compare it, yeah, at least you can propose, yeah, in the future, yeah, what is the benefit, yeah, the benefit, because the RNA experiment is also not really cheap, yeah, it is also a little bit expensive, yeah, but now I think it's not so, yeah. So at least you have to get more what is question, yeah, question that you can offer. So for example, this, this is the in Europe. In Europe, they have a problem. Yeah, they have a problem with the meat quality from the pig. Yeah, from the pig, especially from the boar. Yeah, from the boar, they have like Borthin, yeah, unpleasant odor when people consume the meat. Yeah, and then normally to reduce this Borthin, yeah, people castrated the animal, yeah, since early in the beginning, yeah, 7 day or something. But this in Europe will be banned, yeah, because due to the not related with the animal welfare, yeah. So this Borten is more affected in the liver metabolism, yeah, inside of the body. Yeah, they have a fat, they have a urine or something like that, and then we try to estimate, yeah. Here we measure 100 boar, and then out of the 100 we select 10 who has really high, yeah, and lower Borten, like that. Yeah. So for example, this is the high androstenone. Yeah, this is the one major component or chemical compound, yeah, which is affected to the Borten. Yeah. For example, high androstenone we select more than 1, yeah, microgram, or one, yeah, this the same. And then low androstenone we select 0, more than or less than 0.5. And then out of the 100 we select 10, which is high, yeah, and low. So in the high group we have 2.48 or 248 nanogram, 2000. And then in small or maybe in the lower, we have only 200 or 0.4 microgram or 200 microgram. So this is, we want to analyze transcriptome because we want to see, yeah, we want to reduce, yeah, the animal which has higher Borten, like that. And consumer is not like it. Yeah. So in the future, we hope we can produce, yeah, a pig which is have meat quality with lower androstenone or lower Borten. And then we isolate what is the source of the androstenone. So they have 2, yeah, first in the liver and then second in the testes, and then we characterize, yeah. And then secondly, we try to analyze also from 62 sample from the meat of the chicken, yeah, and then we compare. They have a high unsaturated fatty acid and then low unsaturated fatty acid. So fatty acid is important, yeah, especially now people try to afraid with the cholesterol, with the coronary heart attack or something like that. And then we try to characterize. The high, for example, here is 57, and then the lower is 51. Yeah. So this is why we want to compare, yeah, or to do transcriptome between 2 different conditions. Yeah. And then this the experimental method and workflow, yeah, they have this DNA, yeah, remove DNA and then transcript, convert to cDNA. After that, sequence alignment, and then you can get the sequence. So this is the the flow chart. What is the experimental method and workflow? They have a sample preparation. This is very crucial, yeah, because the first, yeah, quality that will be good, yeah, is depend on the sample preparation. If this quality is bad, so maybe we cannot really guarantee, yeah, the result, yeah, because this is the that we can control it. And then we have to make a library construction, and then also a sequencing. Yeah, sequencing. This is by tools, yeah, facility, and then also how to handle raw data. Yeah. So this is that I mentioned how to get the RNA quality. Yeah, if you take a sample, yeah, from the muscle, for example, from the liver, and then you make sure, yeah, if the quality of RNA. What is the RNA? Yeah, the RNA, they have two indicator. Yeah, they have 18 and also 28 ribosomal RNA. So in this case, at least we can see, yeah, they have two clear band that we can guarantee, yeah, this is more, at least the quality RNA is, we can check it, yeah, by semi-quantitative. Semi-quantitative, it means we cannot determine the score or maybe the quantification, yeah. So but now we have the facility how to quantify RNA, yeah. The quality can be measured from the 0 until 10. The 0 is very bad, yeah, of course, and then 10 is excellent, yeah. But the normal that we can sequence, yeah, the sample, normally at least you have 6.0, yeah. RIN, it means RNA integrity. Yeah, you can see here the RNA integrity is 6. Yeah, we found 2 band clear, but they still have a noisy, yeah, from other maybe contaminant or something like that. 7 maybe is more better, but 10, you can see here, 10 is perfect. Yeah, only they have a 2 band here. Yeah, 18 and also 28. No noisy they have, but maybe still. Yeah, but very rare. Yeah. I will show you how we can get the RNA integrity when we characterize the RNA. Yeah, so this at least our experiment, yeah, this score is 7 or 8, yeah, 7. But we can see, yeah, at least acceptable when we want to do RNA sequencing. Yeah, so this also, yeah, this is the RIN value greater than or equal 7.0. Yeah, 7 at least. The quality at I forget is 7. 6 is okay, hold, it's mean we can propose, yeah, to sequencing, but below 6 normally is fail, yeah, like that. So this is our sample from the 2 species. SU we analyze from the sheep. Yeah, you can see the RNA integrity is 8 or they have a 2 hold, but this we can sequencing. But you can see, yeah, the CS, this is from the chicken, yeah, the quality is almost perfect, 9.8 and 9. Almost 10. Yeah. So if we know the quality of RNA is very good, yeah, we can guarantee, yeah, when we come or when we move to the sequencing, the result will be good also. Yeah, like that. Okay, so and then analyze. Maybe this is the method how to analyze, and the method they have PCR, yeah, and then like I mentioned, after we sequence and then we get a lot data, and then how to handle, yeah, they have some kind of analyze, yeah, differential express gene, and then also they have some enrichment analysis. Yeah, we make a classification. Yesterday we talked about the clusterization. Yeah, this is important. Which one the family gene, yeah, inside of the clusterization. Yeah, and then summary data production from the bigger data, and then we want to make a summary which information that we have to provide. We have to provide in the data, yeah, like that. This raw data, and then we have to make a QC, quality control, yeah, for the result of the sequencing, and then also other, yeah. This, for example, yeah, this is the raw data statistic that we can see, yeah. Yeah, normally they has the data like that, yeah. This is the best quality. The good quality, it should be in the green, yeah, in the green, yeah, in the green here, yeah. But this maybe you can see they have a lot, yeah, distribute in the orange and also in the red, yeah. So this yeah, this is our sample normally, but you can see this is, we still found the data located in the red color, but here perfect, yeah, here is perfect because the data is distributed in the almost in the green, yeah. Green is above the quality 20, yeah. But they have a technique, yeah, to at least to maybe to minimize, yeah, the unusual data, and then we can use as a trimming, trimming data statistic. This is before trimming. You can see we still found in this orange, but after trimming, at least you can reduce. You can find all the data location, the sample in the green. Like that. They have the trim data. Okay. So now this is the mapping quality. The mapping quality from the data, and then for example we have two sample low. For example, here we talk about mid, and then we measure the fatty acid composition. They have a low unsaturated fatty acid and then high unsaturated fatty acid. Inside of the group, we have an individual. And then we can see how the percentage of map to the read. It means the sample, when we alignment to the reference, how many percents the success. And then we can see this almost is high, 90%. So only 9.23 is unmapped. So before in 2009, because since it's very rare, using the RNA-seq technology, and also the complete maybe genome also still not like now. The percentage of map is like 50%. But now, since both is improvement, the technology is more cheaper also, and then the people more update also to database, and then now is the quality is become 90% like that. So almost in our case, the mapping our sample to the reference above 85%, like that. So, and then now after we know the step by step how to do, if you read the paper, transcriptome and stuff like that, and then they have a step that I already mentioned, and then how the application of the RNA-seq in livestock, because our background, our field is livestock, so of course I have to more specific to the livestock. Otherwise we give more illustration with COVID is more easier, but now we come to the livestock. So this is the several application of the RNA-seq is already proof in the livestock, like a pig, chicken, cattle, and sheep. So in pig, for example, trait, what is kind of trait, protein, and then also the muscle, and then chicken is fatty acid, and then in cattle, rib area muscle, tenderness, or marbling, marbling quality, like a Wagyu cattle, this is very economically important trait. And then a sheep, they have a flavor and odor. Meat quality trait, like maybe I will explain later. So this actually to give more understanding about the transcriptome analyze. So maybe I will highlight some our work from our home. So we already characterize the, like I mentioned before, the Borten composition, scatol and also androstenone is already, and also we try to explore also, because this is problem in Indonesia. People, some people, they didn't want to consume the meats from the lamb or sheep, because they have a unpleasant odor, unpleasant flavor also, that we call in Indonesia is perengus. So and then we try to characterize what is the component, which is compound to the flavor and odor in sheep meat. And also in chicken, and chicken also we try to characterize transcriptome to compare between meat with high unsaturated fatty acid and lower unsaturated fatty acid. Okay, so I will give you more deeply understanding, which is in our case. So in this case, we know one of component which is affect to the Borten is scatol. Scatol, and then from the scatol we we measure the scatol parameter. Yeah, we measure from 100 bore, and then we select the extreme phenotype, which is high and then which is low. Yeah, the high is scatol above 0.45, yeah, or 450 nanogram, and then the lower is 0.09 or only 90, yeah, 9, yeah, 9 microgram. This is quite wide range, yeah, quite wide range. And then for the transcriptome, you have to choose the different phenotype, the divergent phenotype. For us, yeah, for livestock, maybe sometimes it's a little bit difficult to find out because we have to measure, yeah. But in the different condition like a normal and disease, this is no need to measure it because people already decide, yeah, or maybe from the hospital, from the doctor is already this is disease and this is normal. But for us, for the complex trait and then like meat quality, other, yeah, growth or something like that, we have to measure it, yeah. Otherwise, we cannot get the divergent phenotype. Yeah, and then we get after that. This is the workflow or how to identify the transcript, and then also to find the gene which is influenced to different scatol level, mapping again, sequencing, and then mapping after that, and then we select what is the differential express gene, and then the gene which is influenced in the differential express, yeah. And then we do also the clusterization, and then we validate, yeah, from the sequencing in our laboratory also. And then we validate also in the population, yeah, that we have. For example, in this case, we validate in 100 animals, yeah. So this we summarize. This is because in the step of the RNA-seq, we have to summarize how the percentage read. Yeah, in this case is still less because this is before. Yeah, in chicken is 2019, in this 2013, 61%, 86, yeah, 68%, yeah. Low map 60%, and then high is 80%. But in this case, at least in the range, yeah, before it's 44, like I mentioned, in 2011 when the RNA-seq quite new, yeah, introduced to the community like that. Yeah. So this is the result of the transcriptome analysis. Yeah, this is we have a phenotype low scatol level and then high scatol level. This is very clear, yeah, the difference, yeah. And then inside of the high and the low, we found several constituent elements of gene, yeah, this who make differentiate. For example, in high level, this gene, yeah, for example, ATP5B is higher. Higher is red, yeah. Higher is mean upregulated or overexpressed, and then green is lower expressed. But in the lower scatol, this gene should be lower. So if we want to get the animal with lower scatol, at least you have to make a condition for this gene, enzyme or protein should be lower. If high, this will be high scatol. Who people want to avoid, yeah, avoid this high scatol. But people normally want to get this kind of such meat quality of this, yeah, like that. So, and then we found out the 448 differential express gene, which is 380 mostly overexpressed, and then only 65 down. So maybe in this case, we want to get more, yeah, characterized gene which is have a lower express, because lower express could be associated with the lower scatol. Yeah. So, and then by clusterization, we get the member of this kind of gene, for example, this transferase, yeah, and cytochrome, yeah, cytochrome who dealing with this lower scatol. So this is actually after we found out a lot of gene, and then we make a clusterization, and then we found only several family, yeah, to become more specific, yeah. And then what kind of the data after we get a lot, and then we can classification, yeah. Gene which is related with small molecular biochemistry, we get 53. This is the high. And then related with protein synthesis, related energy production, which is related with the skeletal metabolism. Yeah. So, and then we can see, yeah, we can know what is the functional categorized. Yeah. And then sorry, this a little bit enrichment is already because I have to handle this. Yeah, and then we know also the pathway, yeah, pathway which is related with this scuttle. Why is high scuttle? Yeah, the pathway which is related with this remodeling, for example, epithelial adhesion junction, it is related with factor who affect to scuttle, high scuttle, yeah, and also other factor. Yeah, and then in this a lot of gene, because we know the clusterization, and then we select gene which is related, for example, cytochrome, yeah, cytochrome related with CYP, and then energy metabolism, ATP, yeah, and also this related with transferase, for example, this is phosphoglucomutase, yeah. Yeah, we found out the maybe this candidate gene which is important, yeah, when we want to make a breeding program to reduce the burden, yeah. So, and then we found out also the RNA sequencing, not only the differential express gene, but also we found the difference between too high and low scuttle could be by mutation. Yeah, mutation by which gene? Gene which is related with cytochrome and also with energy metabolism, ATP5B. Yeah, this is non-synonym, and then this is expressed in high, lower scuttle. Yeah, like that. So now if we make link, yeah, from the result and then to the biological function, yeah, so this is in line, yeah. Metabolism of scuttle is start from the gut, yeah. Here they have a lot of component, and then come to the liver. Liver, they have a two phase, yeah. Phase one, it is related with the oxidation. The oxidation process is more element or more constituent related with the cytochrome and also energy metabolism. And then phase two, which is this come for the final process, yeah, could be produce fat or maybe excretion in the urine. They have a sulfa conjugation, yeah. This is SUL, and also this is related with the ion transport, yeah, like that. And then from the metabolism liver, and then we can make what is summarize, yeah. They have a two phase, oxidation and then conjugation. Phase one, which is we found the gene which is related with the oxidation, they have here, and then also the phase two, where is a conjugation, they have this kind of gene, yeah. We know at least the high scuttle because of the energy production gene is high, yeah. This also ion transport gene also high, yeah. How about the lower scuttle? The lower scuttle, at least, yeah, when we want to get the lower scuttle, this kind of gene energy metabolism should be lower, and then the ion transport also should be lower. Yeah, so this is the method of how to manipulate in the next, yeah, in the next experiment or in the next application. Yeah, and then finally we validate, yeah, the gene or maybe the element, the enzyme or the protein that we get, yeah, and then we associate with the animal, yeah. 100 animal, we already have the scuttle, yeah, value, and then we correlate, yeah, how the marker our gene that we have, and then we associate with the individual scuttle. And then we found, for example, here, yeah, for the here For example, for this keratin gene, at least we found, they have 3 genotypes, TT, TC, and CC. This is the mutation. In keratin gene, they have a mutation from G to A, from guanine to adenine. So in the condition of the homozygote GG, they have a high scatol. In the heterozygote GA combination is middle, and the AA, the adenine combination, it is low. So in the application, maybe after validate in mostly several population, at least we can select animal which have AA genotype, maybe could be proposed has a lower scatol level. So in the breeding program later, if we want to produce pig meat with the lower scatol, so you can multiplication animal which have AA condition. If you have AA and then multiplication with AA, the result will be AA. If the AA, it means the lower scatol, like that. By which tool? At least you can use this candidate marker. Keratin A, keratin A, like that. So this is one kind of the association study. So this also, because this is mRNA, and then focus with the exon which is the region, the translated region, and then we can see also which one is the inside of the gene to make a variation. The variation not all in the gene, but they have in the exon, for example, here in the exon 12, who make a difference in exon 12, not all in the gene. So from the RNA sequencing, we can know more deeply until the structure of the gene, and then in which structure the differential between two conditions is inside, like that. So this is the conclusion, number candidate gene related with metabolism scatol identified, and then from the RNA sequencing also we can identify, related with the biological function, the constituent or gene which is involved in the phasal condition, and also in the conjugation, which is related with the metabolism of scatol. And then we find also the gene which could be potential for candidate marker or scatol in the bone. This may be the more illustration that I give to you. This is the application, how to make a model, androstenone and scatol, in this, how to control, if you want to get lower androstenone, at least this candidate marker should be lower, and the liver, the androstenone, this is the gene should be lower, and but in this scatol should be up, and then we can see other can excrete it, and then this will be accumulate in adipose tissue. That's maybe the illustration. Other we already characterize also in sheep, like I mentioned before. They have a problem in mutton odor, and then they have two components actually, maybe I briefly here because no need to make a detail. They have two components, methylphenol and also scatol. Same like in Borten, they have also scatol in meat, So, and then we have a different divergent phenotype from 30, and then from 30 we select 3, which is divergent. For example, here only G1, and then we compare here, 1.6, 0, 0, 1, here 0, 1.6, The same, the same. This we found out the map, 85%. This also we found between high and low. We can see what is the difference. Clusterization, and then gene also related with the mutton odor flavor. And then here finally we get the genotype like before. which one is more high or maybe lower with the androstenone, yeah, and then we can select. For example, this methylindole, yeah, in this term of the TT, yeah, TT genotype is lower. So it means in the next breeding program, maybe this, if you keep this animal, will be produce a lower mutton odor flavor. Yeah, I think that's the final conclusion. Experiment produce a high dimensional data, and then in order to perform robust statistical analysis, the methodology must be carefully designed. Like what is the equation, and then RNA-seq provide high resolution transcriptome in order to get more high output, yeah, like a differential gene expression annotation and also enrichment analysis. And then RNA-seq also discover new transcript like exon Jusaic and also single nucleotide polymorphism, which can be used as a potential marker for assisted selection. Yeah, I think that's all from my talk. Thank you, and then I give back to moderator. I did. I back to you.
Okay, thank you very much, Prof. Asaf, for a nice and very interesting presentation. And we are going to the discussion session, yeah. So there's already three participants who deliver a question. So while waiting the others, so I would like to invite Miss Mita Sari from Mula Warman University. Perhaps you would like to ask directly. I already write down the question on PowerPoint, but if you don't mind to ask directly to Prof. Asep, please, time is yours. Mrs. Mita Sari from Mula Warman University. Okay, perhaps there is some problem with the audio or something. So Prof. Asep, I will uh, uh, deliver the question, yeah, from Miss Mita Sari. There's a three question actually from Miss Mita Sari, and then from Zilan Aziza, and from Mr. Rafi. The first one is from Mita Sari. She asked about how long. Oh, wait a minute. Okay, wait. Okay, share screen. Okay, perhaps you can read. You can see the question, yeah, from the here, Prof. Asep, from Mita Sari. How long we can get the result with RNA sequence application app? How many sample in population we need in livestock? That's the question from Miss Mita Sari from Mula Warman University. Thank you, yours, Prof. Asep, to answer this question.
Okay, thank you, Ibu Mita, ya, Ibu Mita Sari. I don't know, maybe your background is animal science or maybe forestry. Normally, Mula Warman has a strong with the forestry. Okay, so this is practical question. Okay, so the result of the RNA-seq is normally when we give to the sequencing platform, normally around 6 weeks. Yeah, 6 weeks is normally we can get the result, but depend on the, like I mentioned, the important thing, we have to really carefully about the preparation of the sample. Sample should be high quality, yeah, like a RIN integrity above 7.0. Yeah, if not yet you get this RNA integrity or maybe the qualitative, like we see in the electrophoresis gel, not very clear, yeah, or maybe still not fine to bend, better to repeat, yeah, until you get the good quality. Yeah, so after get and then send for the sequencing, normally 6 weeks, yeah, you can get the result. And then how many sample in the population we need in the livestock? So normally for one kind of sequencing, they have a 12 line, yeah, 12 lines, yeah, 12 line. 12 line is maximum. Yeah, if you have a 2 phenotype, maybe you can maximize. Yeah, 6, for example, one condition and then 6 other condition. Yeah, for example, one in our case, for example, high Borten and then 6 is lower Borten. Yeah, before in 2013, I sequencing 3. Yeah, since it's rare. Yeah, before 2009, especially in yeast. Yeah, only one sample because it's very expensive. But now I think you can maximize. Yeah, 12 maybe is good because it's quite enough for technical biology. Yeah, at least 3 is okay, but normally some people, why only 3? Yeah, 5, yeah, is okay, and 6 is best. Yeah, you can maximize. Normally 6 versus 6, yeah, is enough. Yeah, because the price of the sequencing the same. Yeah, if you sequence only 2 sample, the same like 12 sample. Yeah, the different thing, each sample, yeah, you have the what is the sample preparation. Yeah, it's the sample preparation. But the sequencing, one sequencing is the same, 2 or 12. Yeah, I hope I can answer the question.
Okay, thank you, Prof. Asep. The second one is from Zilan Aziza, IFB University. Zilan, would you like to ask the question directly to Prof. Asep, please?
Thank you, Mr. Aditya, for the
Okay, sir.
opportunity. Okay. Hello, Mr. Asep Gunawan. My name is Zilan Aziza from IPB University. I'm from Nutrition and Food Technology major. Based on what I know, RNA is unstable and very easily affected by the condition of the cell environment. I want to know what is the good condition of the cell environment to do RNA sequence or something in the transcriptome analysis, maybe using PCR or something, I don't know, and the probability of a failure. Thanks in advance.
Hello, Professor.
Yeah, okay. I hope I can get your question, Zilan. Yeah, okay, good. So RNA unstable, very easily affected by condition, environment. What is the good condition of cell environment to do RNA sequence? Okay, so good. Yeah. Since it's not stable like DNA because DNA is double helix. Yeah, and RNA is only one helix. Yeah, so you have to make sure, really free from the contaminant. Yeah, from the contaminant. At least like when you make an extraction, you have to really normally, in when I study in Germany, they separate between DNA lab and also RNA lab. Yeah, RNA lab is normally they close. Yeah, the pipetting that used for the RNA cannot use for other purpose. Yeah, RNA for PCR or something like that. Only for the RNA. Yeah, so you have to put in for example, in one box. Yeah, all pipette for the RNA, and then the band or the table when you work for the RNA should be clean. Yeah, should be clean every time. Yeah, every time like that. Yeah, if DNA is not like that. Yeah, but if like that is also more good. Yeah, but at least for RNA, should be different room I suggest, and then different facility pipetting should be alone, and also the surrounding of the table on everything should be clean. Yeah, like that. Yeah. And also when you extract and then you what is the isolate, the condition should be clean. Yeah, so this is to avoid the contaminant. And then what is other Pak Edit? For RNA, for RNA sequencing, if you already get work with the clean, and then you sure everything on your control. And then when you run to the electrophoresis, and then you get 2 band clear, yeah, 2 band clear, yeah, because bioanalyzer normally we didn't have, yeah, but at least you can measure electrophoresis and also nanodrop. Yeah, if the nanodrop is like you have what is normal distribution, yeah, so maybe this is good. Yeah, this is good. Yeah, they have a comparison between DNA and RNA and DNA quality. Yeah, 280 per 260 like that. Yeah, should be below 2 actually. They have a measurement. Yeah, and then of course if you have this 2 band, yeah, if you didn't have a bioanalyzer, and then yeah, you can send it. But if you have bioanalyzer, is more better because you can measure. Our DNA is already has above 7.0 or below. Yeah, if below, maybe yeah, at least you can repeat again, yeah, until you get the maximum. And then the handle, the important handle for the RNA, better when you collect the sample as soon as possible. You have to analyze, to extract directly. It's better from the fresh sample. Yeah, from the sample. When you, for example, slaughter the animal, and then you collect, for example, tissue sample like a brain, like a liver, and then better you collect and then you extract directly. Yeah, but if you, for example, storage in the refrigerator, otherwise we put in -80, maybe they have some degradation. Yeah, so this is the crucial point. Yeah, I hope I can answer your question, Chilan.
Okay, thank you, Prof. Asep. The next question is from Aldi, University of Gajah Mada. Aldi, would you mind to deliver the question directly to Prof. Asep, please? Time is yours. For Aldi? Okay, if it's not on the room, or we can read the question, Pak Asep. Okay, this is the question from Aldi, Pak Asep. Prof. Asep. Good afternoon, Prof. Asep. I'm Aldi, because she also has doing his undergraduate thesis, yeah, regarding about the molecular marker selection. How to downstream research from molecular genetics to ruminants, which tend to be more difficult to apply, or even less applied than to plants or poultry. Related to the use marker assisted selection, besides the research of marker genetic is more expensive to apply in breeders, but we know that using mass is more effective and can cut more time of research for many years. Please, pass up, time is yours.
Yeah, it's difficult question, yeah. Okay, but I will try to answer. How to downstream research from the molecular genetic to ruminant, yeah, which tend to be difficult to apply. Yeah, okay. So yeah, normally people not really interest to what is to know, yeah, the at least to improve, yeah, genetic improvement, yeah, for the animal, because this is of course not really can be find what is solved, yeah, in the fast way, yeah, because normally it's long term, yeah, long term, yeah. For example, when I characterize this in ruminant, in sheep, so I try to improve from 2016, yeah, from 2016 until now, yeah, and then we found, for example, the marker 2019, yeah, at least after 3 years, yeah, and of course it is, if you provide by yourself, maybe it's impossible, yeah, impossible, yeah, because maybe you have to what is to, yeah, to spend a lot money, yeah, but by collaboration, by project funding, yeah, from the country, yeah, from the ministry, maybe we can improve it like that, yeah. So from that we characterize, yeah, and then we try to apply, yeah. This is, yeah, from year to year, yeah, like we characterize from the 7 population. Now we already 7 population, at least 400, yeah, 400 sheep, yeah, we validate this marker, yeah, And then we also has now the selected population. Yeah, selected population still less than 100. Yeah, still less than 100. But this is we hope this is the right individual that we want to propose, yeah, for the good genetic improvement in terms of meat quality for ruminant, like that. Yeah, we still have a roadmap. We still write a roadmap until 2030. Yeah, 2030. Yeah, and 2030. Yeah, we hope we already get the population by producing or using marker assisted selection. Yeah, I think that I did. Yeah.
Okay, thank you, Prof. Asep. And then before we are going to the next question in the chat, there is somebody who raised their hands. Mr. Adnan Ahmed from Bangladesh, your raised hand. Times is your Mr. Adnan to ask directly to Prof. Asep. Times is your, please.
Hello, sir. Can you hear me, sir?
Yeah, it's okay.
Sir, actually, sir, you told about enrichment analysis and differential gene expression. I want to know, why do you perform enrichment analysis in case of finding a novel gene? Or what is the function of gene ontology before performing enrichment analysis?
Where is the question?
Yeah. Okay, so I hope I can get your point. So this is related with the enrichment analysis to find the novel gene. Yeah. So before we do enrichment analysis, yeah, maybe previous study already mentioned or reported the improvement, yeah, could be in meat quality or something like that, but they approach using a single gene. Yeah, single gene. For example, they characterize one mutation, this gene, for example, cytochrome A1, for example, in this. Yeah, when we do enrichment analysis, yeah, we can get like a comprehensive clusterization. Yeah, clusterization. For example, people who already get CYP A1, this is related with cytochrome. When we get the enrichment analysis, we can get a lot, yeah, not only one, but maybe different variant. Yeah, different variant. Maybe could, in our case, people mention cytochrome A1, but in our case, we found, for example, cytochrome B5, for example. So this is that we speculated, this gene is still in the related family, but novel in our case, in our sample that we sequence. Yeah, like that. Yeah. So then we found, yeah, since we know the biological function, for example, scatol metabolism in the liver, they have a two-phase, oxidation, and also they have a conjugation. So the gene or maybe the element is only related with two conditions, yeah, cytochrome and sulfur and conjugation. So to find the novel gene, yeah, the different variant or maybe the different family, yeah, could be fine. Yeah, so that's why we say we can found the novel gene. And for the gene ontology, yeah, they have a several software that you can use. Yeah, they have a like IPA, immunity pathway analysis, and also they have a like a maybe free access that you can use as David. Yeah, David. It's mean I forgot David visualization. Yeah, and David. Yeah, David. Yeah, this is also for annotation. Yeah, for the clusterization, like that. Has my answer answered your question or not?
Yes, sir. Thank you.
Yeah, good.
Okay, thank you, Prof. Asep, for the answer. Next
Yeah.
question is from Mr. Rofi, IPB University. Mr. Rofi, would you like to answer, address the question directly to Prof. Asep Gunawan?
Yeah. Sorry, for who is from
Rofi.
before? Yeah, Rofi. So this is maybe the last that I mentioned. This is David. I forgot the
Okay, yeah.
abbreviation. Yeah, this is pre-access that you can make enrichment analysis. David is database for annotation, visualization, integrated discovery. Yeah, this you can read it. Yeah, I think is that maybe if I did.
Okay, thank you. Perhaps Mr. Rafis have a trouble with the audio, so we can read the question. Mr. Rafi, Professor M. Rafi Bella. One question, the 5 gene markers could appear in the existing composite ship or only from selected breed. It's regarding about the breeding of sheep in Indonesia. Indonesia will have premium land. One question, the 5 gene markers could appear in the existing composite ship or only from selected breed. Thank you.
Yeah. Okay, since gene build up our constituent genome, yeah, the gene, yeah, of course could be appear, yeah, in term of different breed condition, yeah, either in the composite, in the Japanese fairtail, in the Japanese tail, in the Garut sheep or other. For example, if we found the gene, for example, cytochrome, cytochrome 5 CYP2A, yeah, this gene, yeah, will be show also in species of the sheep, seems the same, yeah, seems the same, but the level, the quantity, the transcript of the gene, yeah, among the breed, yeah, maybe could be different, yeah, could be different, yeah, could be different. For example, maybe the important thing, the easy thing, like a myostatin gene, yeah, myostatin gene is expressed in the Belgian blue, yeah, in the Belgian blue, yeah. Pak Edit is more know about the cattle. Whether myostatin also present in the Bali cattle or maybe in the PO, yeah, they have also myostatin, yeah, but the expression, yeah, the mutation that we found, maybe this different with the Belgian blue, yeah, the expression is different. The same also like a marker that we found, a 5 marker, yeah, of course they has also in other sheep breed, yeah, but the transcript, the quantity of the transcript, maybe it could be different, like that, ya Pak Rofi, ya.
Okay, Pak Rofi, enough. Okay. Yep.
Thank you, Kang Edit.
Yeah. Okay, we are going to the last question, Prof. Asep, from Muhammad Mursid Mustafa from Bangladesh. Mr. Mustafa, would you like to address the question directly to Prof. Asep? Okay, there is no response at all. So this is the question, Prof. Asep, from the, pardon? Yep. From Muhammad Mursid Hassan Mustafa. It is nice presentation, Prof. Asep. How long we get the results with RNA sequence in diary? Perhaps you have an experience or something, Prof. Asep, for this question. Thank you.
Yeah. Okay, maybe it's like first question, but maybe more specific in diary. Yeah, normally for the diary, it is not like tissue cell, yeah, because milk, yeah, normally milk, and also of course when dairy cattle, it's very expensive and not possible to slaughter, yeah, because they can produce for the milk production. So they have several techniques that normally people use for the liquid sample, like a semen, yeah, semen and also milk, yeah. They have a technique, yeah. For example, in our case, we already characterized with the semen quality, yeah, semen quality, yeah, and then they have a specific kit, yeah, to make a cold pulse, to make a cold pulse, in the refrigerator like a meal or something like that. And then after that, we isolate, we isolate to make a better solid pulse, So at least for the dairy, for milk, they have one step before, different like muscle when we slaughter and then directly to isolate. For this we have to treatment before, to make a cold, to make a solid, and then after that we can isolate, using maybe a specific protocol, milk for milk and also for cement. And then after that same, we measure the quality of RNA and then the quality of RNA integrity, and then after that you can sequencing the sample like that. That's maybe what I did, and then the result is the same. If everything is fine, normally 6 weeks. 6 weeks is already 6 weeks already came. With the result. Like that. And then what kind of the result that you have to expect? It is important also. For example, I want to get this differential express gene. You want to get the enrichment analysis. Maybe otherwise you can also analyze. It's also that you have to see what the question before you do. It is important. Like that. Okay, like that, Pak Edi.
Yes, thank you, Prof. Asep, and Mr. Mursid already write on the chat that he said thank you, sir, for the answer. So it's mean we are going to the end of the session, Prof. Asep. Thank you very much for your lecture, for your explanation, also for the time for the discussion. And we do apologize if there is some question that are not yet read or not yet discussed with Prof. Asep. Probably you can send the question directly to Prof. Asep because there is an email, I think, on the slide, or you can deliver the question to the committee, and then Prof. Asep will ask the question. Again, thank you, Prof. Asep, for the interesting topic, And before we are close this session, we would like to give you a certificate of appreciation from the committee. Okay. On behalf of the committee, head of department, and also the dean of Faculty of Animal Science, we would like to deliver the certificate of appreciation for Professor Dr. Asep Gunawan for this very nice and interesting lecture in this summer course program 2021. So
thank you, Pak Edi. Thank you, the committee.
Okay. Again, thank you very much. Is there any announcement from the committee? Not yet, So before we leave, I think we can take a picture for all of us. Pak Mas Dimas or Mas Bang Arman for the take lead to take a picture all of us. Please, the committee. Okay. Thank you very much, the committee, and again, Pak Asep, thank you very much, and also for the all participants that joined with for all session today, from the first one and the second session. We do hope that we are always in a good health, Stay healthy, and then we'll see you tomorrow for the third day of our program, Me as a moderators would like to appreciate to the committee, to the speakers, to all the participants, and apologize if there is something or probably something that are not still not good enough, yeah, during this today's session. Okay, stay healthy for all of you, and then goodbye. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullah. Thank you Prof. Asep. Thank you Prof. Aminul. Viva Engineer, Viva Veteriner. See you tomorrow.
Thank you very much.